Welcome back to day 2 of EPA's second annual conference on new approach methods for use in chemical safety testing. My name is Anna Lowett. I'm the Senior Science Advisor at EPA's Office of Pesticide Programs. Today, Maureen Gwen and I will share duties to moderate today's session. Just like yesterday, we asked the participants to use the Q&A box for questions to the speakers and to the EPA team. Just like yesterday, we will do our very best to answer those questions either on the audio or in written form through the Q&A box. In those questions that we are not able to get to during the day, you will get a written response back at some point in the near future. Keeping in mind, for those of you submitting anonymous questions, we don't have a way to, we'll try to get those questions answered because we won't know how to answer them directly. Yesterday was fantastic. We had a wonderful attendance, some really fantastic presentations, and anticipating another very active day. So the Q&A box was quite active yesterday, and so we're looking for another active day in the Q&A box. Just to remind everyone that the conference presentations and recording will be available soon on EPA's website, and all participants are muted to allow for the speakers to present without too much interruption. So without further ado, I want to introduce Alexandra Dunn, who is the Assistant Administrator for the Office of Chemical Safety and Pollution Prevention since January of 2019. And before that, she served as a Regional Administrator for EPA's Region 1. And we're very lucky to have Alex here with us today. She, just like the Administrator yesterday, Alex is a huge proponent of the 3 R's, the reduce, replace, refinement of animal testing, not only because we should make sure we're using the animals the best we can, but also because Alex understands that moving to the 3 R's makes our science assessments they're more scientifically supportable. We can be more efficient with them. So we're lucky to have Alex here this morning. So Alex, I'll turn it over to you, and thanks for being here.
Thank you so much, Anna, and good morning, everyone. It's incredible to be virtually with over 300 of you at this very moment, and throughout the day, we have nearly 900 people registered from around the world. What an incredible coming together on such an important issue. I know that meeting virtually is not our preferred way to get together, but it allows us all to navigate the current public health emergency during these very challenging times. And I want to thank you all for embracing this platform and format and engaging with us in this way. Notwithstanding events, we have to keep moving forward on our new approach methods. And Anna is absolutely right. I am very passionate about this agenda for the EPA and for, frankly, the country and the world. I know, as you heard from Administrator Wheeler yesterday, he has really challenged us by setting some very ambitious goals for this agency with respect to prioritizing our efforts to reduce animal testing. His September 2019 directive built on a lot of progress that this agency had been making over many years to reduce our reliance on animal testing. And under his memorandum over the next 15 years, EPA will continue to improve the science that we rely on for our decision-making and work towards eliminating the use of mammals in testing where it is scientifically proven that alternatives are available. This conference gives us that opportunity to hear from many of you, from scientists from around the world, on the state of science with regard to reducing the use of animals in toxicology studies. EPA is working collaboratively with scientists from academia, government, industry, and non-governmental organizations. Because the science and policy issues surrounding animal use reduction is complex, these multi-stakeholder activities, like this gathering, are so critical to us achieving our shared goals. I know that yesterday you heard from Drs. Rusty Thomas in our Office of Research and Development and Gino Scarano in this office, the Office of Chemical Safety and Pollution Prevention. Rusty summarized the EPA New Approach Methods work plan, and Gino described the progress that EPA has made to use NAMs to help implement the Toxic Substances Control Act. So I thank them both for that presentation. I know that you also heard presentations from stakeholders outside of the agency. These incredible presentations highlighted recent scientific advances in the development of new approach methods and how these advances are helping to achieve the goal of reducing the use of mammalian studies. So as Anna said, we are off to a great day today with a robust and rich agenda. Today, we will hear from the Office of Research and Development's Dr. Chad Dizenroth about the development of an innovative new approach method for incorporating metabolic competency into high throughput screening assays, which many have cited as a weakness of this type of new approach method. In addition, you will hear 2 case studies, one from EPA and one from Unilever. These case study presentations will describe the use of computational or in vitro approaches on developmental neurotoxicity, next generation risk assessments on cosmetic ingredients, and threshold of toxicological concern. As you heard Anna say, if you have any comments or suggestions for the agency on our efforts to reduce our use of laboratory animals, please engage in the conversation today through the chat box, and remember to use our email box, NAM, N-A-M, @epa.gov. I really hope you enjoyed today's presentations, and we very much look forward to continuing to engage with the global scientific community and the public on these critically important issues. Thank you so much.
Okay, thank you, Alex. I don't see any questions for you in the Q&A box. Give it a couple of seconds in case any pop up while we have your time and attention, because I realize you won't be with us all day before we turn it over to the next presentation. Give it 10 more seconds. Okay, so let's just get going on the technical presentations for the day. So the organization of the technical presentations will be very similar to yesterday. We will have 3 presentations and then a 15-minute break, followed by 3 more presentations. Each presentation will be approximately 22 to 25 minutes, followed by a Q&A session moderated by Maureen and I. So if you have questions for the speakers, what we ask is that you type the speaker's name next to the question in case we do get questions out of order, we know the right person to respond to them. And if you have more generic questions for the EPA team, we'll handle those also. Okay, so without further ado, so we want to welcome Chad Dizenroth, cell biology at EPA's Center for Computational Toxicology and Exposure, and he's going to talk to us about the estrogen receptor transactivation assay. So welcome, Chad, and enjoy your presentation.
All right. Thank you, Anna, and thanks to all the organizers. I'm humbled by the invitation to speak today, and looking forward to talking a bit about our work to retrofit high-throughput assays with metabolic competence. All right, so you heard quite a bit yesterday about the 5 pillars or objectives of the EPA New Approach Methods work plan. Certainly the case study we'll be describing today fits within the context of all 5 of these, and in agreement with a number of the speakers yesterday, I agree that you can't really do one without doing the rest. But nonetheless, today we'll focus on the development of NAMs that fill critical information gaps. And so some examples described in the work plan for those information gaps include inadequate coverage of biological targets within in vitro screening assays, limited capabilities to address tissue and organ level effects, lack of robust IATAs, and of course what we'll discuss today is this minimal capability to address xenobiotic metabolism within in vitro test systems. So also noted as part of that work plan, the work we're describing today is housed under a 4-year strategic research action plan, specifically the chemical safety for sustainability plan. And within that, the work is described in the high throughput toxicology research area, where of course we're looking to develop new methods to incorporate metabolism. The work is described, that I'll be talking about today, is described under 2 products. I'll primarily be focused on this product 151, which is the development and application of the alginate immobilization of metabolic enzymes method, or the AIM method. And then I'll just touch briefly on some of our efforts moving forward to try to adopt the principles of that method to a more automated bioprinting approach. Before I move on, I'd just like to acknowledge all of the contributors who have contributed over the years to this project. Specifically, we'd like to highlight Danica DeGroat's efforts, as well as Steve Simmons, as well as our collaborators at Unilever. All right, so there's a number of areas you could start when you look to the needs for incorporating metabolism into high throughput assays. The 2007 National Research Council report seems like as good a place as any. And some key questions that were put into that document for addressing metabolism. There are some interesting quotes in there. One was, one of the challenges of developing an in vitro test system to evaluate toxicity is, of course, the current inability of cell assays to mirror metabolism in the whole animal. And the charge question that they put forward was to ask, how can adequate testing for metabolites in high throughput assays be ensured? And the recommendations coming out of that were to screen using computational approaches where possible, as well as conduct limited animal studies that focus on mechanisms and specific metabolites. But of course, with the efforts to eliminate animal testing, we can ignore that part, but possibly still focus on mechanisms and metabolites as part of the in vitro work. There's also international recognition of this need to incorporate metabolism, particularly when it comes to evaluating endocrine disruptors. And so even a meeting nearly 2 decades ago, it was noted that it was necessary to consider and preferably incorporate metabolism of compounds when considering development of in vitro tests, particularly to reduce the risk of false positives and false negatives. Right? So then they go on to suggest that tests to detect endocrine active substances with metabolism are ongoing, but that the eventual need to combine the outcome of these developments will be an important component of the development of each field. In recognition of this problem, the EPA, in partnership with the National Toxicology Program and the National Center for Advancing Translational Sciences, put out the Transform Tox Testing Challenge in 2016. This was a call to all external scientists to identify innovative solutions to retrofit high throughput assays with metabolism. And this challenge worked through a number of stages where the first round identified 10 candidates who developed proof of principle technologies. 5 of those were awarded additional funding to do a deeper dive into their technology and evaluate and characterize their test systems. And then the third step was intended to then, pick one of those technologies and move towards a robust commercialization of that application. Parallel efforts within the Office of Research and Development were made to continue to develop these metabolic test systems. As reported in this paper that came out last year, the Next Generation Blueprint of Computational Toxicology at EPA highlighted 2 particular approaches. One is the extracellular approach, this idea that you can take chemicals, put them into an in vitro test system that simulates liver metabolism, and then apply that both parent and metabolites to some assay that's representative of a specific target tissue. Converse to that, you've got the intracellular approach. This is a concept where recognizing that the liver is not the end-all and be-all of metabolism, and that there's a number of target tissues that have their own respective metabolism. The idea was to be able to introduce specific types of enzymatic activity directly within bioassays and then detect the outcome. So the combination of the two presents an integrated strategy to model in vivo bioactivation, right? So the intracellular approach is what we call in the mRNA transfection method. This was developed by Steve Simmons. And what it allows you to do is transfect the cell-based system directly with mRNA transcripts that code for some enzyme or enzymes of interest, right? This allows you to bypass the need for a DNA template in order to code for those transcripts. And what's nice about that system is now it allows you to really control the payload of the system, right? You can define the abundance of the transcript that you're putting in there. You can define the composition, right? And to kind of put this to the test, Steve focused on liver metabolism, where he picked 10 most common or abundant CYPs within the human liver and expressed them either in a combination that was reflective of a physiological balance within the human liver, or expressed, in this case, a single CYP alone, CYP2C9. And he compared the activities for metabolism of diclofenac across kind of the three strongest in vitro test systems for liver metabolism. So you're looking at HEPA-RG, which is a liver cell line, sandwich culture of primary human hepatocytes, and then what's considered the gold standard suspension primary human hepatocytes. And he's able to demonstrate fairly high activity by using this mRNA payload transfection method. And then he could screen across a number of compounds and, walking down individual CYPs to demonstrate that, in this case, aflatoxin B1, metabolism to the 8,9 epoxide, induced cytotoxicity as a result of biotransformation, right, and did so at different potencies. So the method we'll be talking about today is, again, the alginate immobilization of metabolic enzymes, or the AIM method. This is a custom pillar lid-based approach, where we've got these lids that are fabricated to have pillars that go directly into a standard 96-well or 384-well microplate. In order to capture that first-pass liver metabolism, we're using a very common reagent in the in vitro metabolism world, which is hepatic S9 fractions. For those unfamiliar, those are coming from tissue liver homogenate, right? And they contain all the enzymes necessary for phase 1 and phase 2 liver metabolism. So part of the problems with S9 in terms of adding it directly to a cell-based assay is that it is noted to cause issues with cell viability, as well as in some cases causing technical artifacts within the assay. So in order to alleviate that issue, the S9 fractions are suspended in alginate hydrogel, right, where, as you can see over here, they're directly encapsulated on the end of those pillar lids, right? So once it's immobilized, then you can put that down into your microplate where you've got some parent compounds of interest, and you can run its course in order to produce metabolites, right? So in some ways, we think of this as a liver on a stick. All right, so we are using S9, very common test system, but of course, still needs to be validated. Once it's been immobilized within that alginate, it's not clear that it's still going to have the same type of enzymatic activity. And so that was the purpose of the series of experiments, where picking a number of reference substrates for a diverse set of CYPs within the liver, we're able to demonstrate that you could see depletion of the parent compounds, accumulation of their respective metabolites, and that this was a dynamic system that you could monitor over time, right? So in this case, we're able to choose a 2-hour incubation time point, kind of a way to maximize metabolism within the system. And something that's also worth noting here is that, in order to get S9 fractions to work, you really need to add cofactors in order to optimize the activity of specific transformation pathways. So in this case, this particular system has only been optimized for the phase 1 CYP activity with addition of the NADPH, and it does not contain cofactors for these phase 2 groups. All right, so we've got a metabolism assay, right? So the question is now, if you combine this with an actual bioassay, how do you do this, and what sort of results do you produce? And so this is the case study I'll describe in brief here today, where we took 63 chemical compounds, pushed them through this AIM metabolism assay, where we evaluated both activity of parents and metabolites simultaneously. Those were then screened in an estrogen receptor transactivation assay. That data was analyzed in the ToxCast pipeline in order to produce all the curve fits. Those curve fits were evaluated for uncertainty quantification using this ToxFood R package. And then that data was then analyzed to rank order metabolism-dependent bioactivity as it relates to estrogen receptor transactivation. So some highlights coming out of this study is that it demonstrates reprioritization of hazard based on metabolism-dependent bioactivity. This demonstrates utility of the AIM method for identifying false negative and false positives within a specific target assay. And also, in several cases, showed enhanced in vivo concordance with the rodent uterotrophic bioassay, which is, of course, the gold standard for in vivo estrogenic testing. All right, so a whole lot of details here, mostly just for those who are interested, but specifically with respect to the assay design specifications, we're taking a look at the VM7 Luck 4E2 assay. This is formerly the BG1 Luck 4E2 assay of OECD test guideline 457. A couple things to note here, again, is that the assay is run with and without metabolism. The dose spacing is a little bit different than conventional screening. We use what we call alternative dose spacing, where we're basically stacking the deck towards the higher concentration ranges in order to maximize the enzyme kinetics within the S9 fractions and produce metabolites that may be bioactive. And then the concentration range is also higher for the same reason. In terms of chemical selection, there were 8 ER agonists, 3 ER antagonists, and 3 negative controls pulled out of OECD test guideline 455. And then in order to test the AIM method, we used what we call the Pinto library. And this was derived from a publication in 2016, where the authors basically combed the literature looking for parent chemicals that had metabolites with demonstrated activity for the estrogen receptor that was either more or less active than the original parent compounds. These chemicals were used to develop ER QSAR models, and then that data was then reported in terms of those respective chemicals and their metabolites and the affinity for the estrogen receptor. So we've got 34 chemicals here that we define as metabolism positive, meaning that they've got metabolites generated from the parents that are predicted to be more estrogenic. And then obviously metabolism negative will be compounds that are potentially metabolized but not producing metabolites that are more estrogenic than those original parents. So this walks through a bit of the quality control evaluation. The things I want to emphasize here, again, are the paired plate design, where the chemicals were run in duplicate plates simultaneously in order to evaluate both a metabolism negative condition where the AIM assay is just got pillar lids that are coated with alginate, and then the metabolism positive, where then you're plugging that S9 into those microspheres. One thing to note here is that in order to compare pairwise plates, apples to apples, you got to have some baseline condition that's the same across all plates. And so column 1 for all of the assays was committed to what we call guideline-like test conditions. So the AIM lid that was put on the plate had no alginate, it had no S9, there was no cofactor supplement in the medium. And this allowed us to evaluate the activity of the controls under conditions that were not different between the plates and could be used for data normalization. So we looked across, this is a representation of the controls across all the plates. These are the guideline-like conditions of 17-beta-estradiol, the natural ligand for the estrogen receptor. This is the activity of estradiol under control conditions. All right, so you see a slight drop in activity. Further, estradiol does get metabolized, so that activity is reduced further. Methoxychlor is a compound that is known to be bioactivated to an estrogenic compound. So under the guideline-like conditions and the control conditions, you see fairly low activity, and then bioactivation as a result of incubation with that S9. And then we looked at pairwise across all these independent variables to ask the question, by combining two assays together, are you yielding results that are still within acceptable levels of variability? And so the long and short of that is that indeed, by combining the assays, you do see more variability, but they are within acceptable ranges. All right, so the data coming out after you've done the analysis in the pipeline, you've performed uncertainty analysis with the ToxBoot package. You've now got these data distributions that you've got to do something with. So traditionally speaking, we would look at any specific assay, look at the baseline of that assay, evaluate the noise band there in order to draw some line in the sand, where we could say that any compound over on the right-hand side here, we would consider to be bioactive or a hit in the assay, and anything below that would be something that would not be considered to be active. And that was a perfectly fine way to do it. I think it certainly helps to identify false positives and false negatives within your target assay. Examples of that would be compound number 2 here, where you've got a parent that's considered inactive and then active after metabolism. So that would be considered a false negative, right? Conversely, you've got compound 6 here, where the parent's very active, but it's detoxified or inactivated after metabolism, so it's no longer active. Right? So that would be an example of a false positive. But immediately, as what you might expect and what we notice is that you see a whole host of situations where metabolism moves the bar one direction or the other, and in many cases it seemed like something that would be relevant for evaluating the true biological activity of that particular compound, and thus would be important for prioritization efforts. Right? So to come up with a way to uniformly evaluate all of the compounds within the screen, we decided to take a step back and simply just ask the question, okay, do we think there are metabolism-dependent effects happening within the test system? And then if there are, then we can go back and look at the relationship of that activity to the estrogen receptor transactivation. Right, so that's what you see over on the right-hand side here is an effort to define metabolism-dependent activity, where basically the difference between with metabolism and without metabolism and the associated variance of that observation, anything that's right-shifted here beyond 0 is considered to be significant. Anything that overlaps here is not significant, and anything that's leftward shifted here would be significant, but for the inactivated direction. All right, so to kind of visualize what some of this looks like, here are the results of the positive reference compounds. Bear in mind, each one of these is fully expected to be estrogenic by definition, and that certainly was the case. Here what you're seeing is the area under the curve, which captures both potency and effect outcomes. You've also got a potency measurement here with the AC50s, and then you've got max efficacy here, which is the max effect level. So evaluating these for metabolism-dependent effects, we didn't expect to see anything here, but we did happen to see one pop up here. So this compound ethylparaben appeared to be bioinactivated. And what was nice is then you can go back and look at the curve fits and really, qualitatively make that call for yourself. So in this case, everything in blue is the parent compound. Everything in red is the metabolite or metabolites. And so, we could walk across and see something like genistein, where you see this kind of bump in activity, but it's not enough for us to say that that's a significant metabolic effect. For the most part, the rest of the reference compounds kind of mirrored the effects of the parent. And then you come over to ethylparaben, where you had very high activity of that parent, and of course, that's reduced down to baseline post-metabolism. So this would be something that we would consider to be potential false positive within our assay here. All right, so taking a look at the metabolism positive test set, what's worthy to note is that 85% of the chemicals right off the bat were estrogenic, according to hit calls in the pipeline, right? But of these, 11 of the chemicals did show bioactivation, right? So that's everything that's highlighted up here in red. And then the corresponding plots are over on the right-hand side here. And certainly as you walk through these, it's fairly compelling that everything in red here is post-metabolism, everything in blue is the parent. So you see very significant bioactivation across this series of compounds. All right, going the other direction, the metabolism negative test set. Again, these are compounds that are expected to produce metabolites that are not more estrogenic than their parents. And for the most part, that was true. 43% of these chemicals were still somewhat estrogenic. But what's worthy to note here, there was one compound that was, significantly activated, but more so this group of compounds here that demonstrated bioinactivation. This was a result that we weren't necessarily expecting. So interesting to note that, we can observe activity shifting in both directions. All right, so once you've got that data set across 63 chemicals, it's nice to put some broader context to it and look across other test systems in order to evaluate and compare and contrast the results. And so here's an example where we just put a table together comparing the activity within our AIM-DM7 luck assay and compared it to the TOSCAST ER model, right? Bear in mind, this is the ER model that captures activity across 18 estrogen receptor bioassays. And the scores are reflected on a scale of 0 to 1, where 1 is being the most estrogenic. And we also compared across eutotrophic studies, more specifically guideline-like eutotrophic studies. Right? And so to give you an example here, you've got genistein, which is one of the reference agonists, considered to be very estrogenic according to the TOSCAST ER model, considered to be positive in eutotrophic studies by weight of evidence. This is just looking at hit counts across the parent and metabolites, right? So there's no change in hit calls according to the pipeline. It does show a trend for bioactivation, but that trend's not considered to be significant, so we say there's no metabolic effect. And the hit calls made both with and without metabolism are fully concordant with the in vivo study, so there's no change in concordance, right? So just walking across the another examples here, benzyl butyl phthalate. Bear in mind this is a metabolism negative substance. It does have some moderate estrogenic activity according to the TOSCAST ER model, but it is negative in eutotrophic studies. And so that shift in the hit calls was noted, right? There's also a significant leftward shift or inactivation of the compound that is considered significant, right? And as a result in that shift in the hit calls, we do see this change in concordance, and now by applying in vitro metabolism to this this ERTA assay, we're able to capture this effect that's observed within the in vivo guideline study. All right, so just a couple more slides here. This one is just to point out that, like I mentioned earlier, we do have a 384-well version of this AIM assay, and you know, we took some lessons learned from the method study to change up some of the controls, but we have since used this to screen through 768 compounds in the TOSCAS library, and so that analysis is ongoing, and so stay tuned for that. And then lastly, this project housed within the research action plan in order to adopt the principles of the method, in order to apply them to this automated bioprinting system. Something we would like to do is to remove these lids altogether and have any end user walk up to the bioprinter, fill it with a cartridge of hydrogel and liver fractions, press go on the instrument, print it out into a microplate, and then we're off to the races to screen metabolism-dependent effects. So, with that, I'll thank you again, and I'll be happy to take any questions.
Okay, Chad, you have a bunch of questions. So a couple of them are related from from the same questioner. So let's start with the related ones. So the first one that came in was from Jay Petrick, and he asks why were phase 2 cofactors excluded?
Yeah, that's a great question. Certainly there's an interest in including the phase 2 cofactors. I think in part it was a big hurdle for us to just get the phase 1 cofactors optimized in the test system without negatively impacting the viability of the cells. And knowing that in large part, the majority of xenobiotic metabolism take place through phase 1 metabolism, we consider that to be an okay first step for the method. But we're fully in agreement that, you know, it'd be very important to have all phases of the liver metabolism present as much as possible.
Okay, and Jay has a follow-up question. How does one exclude the possibility of over-prediction of estrogenic activity if detoxification by phase 2 is not included?
Yep, I would say certainly based on this data set alone, you can't make that determination. That would be something that we would, you know, simply have to follow up on and make some informed decisions on based on the chemical characteristics and expectations for metabolism. Right? So for us, without actually generating that data, it'd be hard to make a call at this juncture as to the effects of phase 2 metabolism on any of the compounds that we're demonstrating here.
Okay, thanks, Chad. So moving on, a question from Atish. How do you account for solubility differences in plus or minus S9 conditions, and how does it impact the interpretation of the results?
So I think the expectation is that the S9 is already solubilized when it's put into the alginate hydrogel. Once it's embedded in that alginate, then the alginate gets cross-linked, and so you've basically got it locked up inside a matrix or a cage, right, within that microsphere. And so our real concern there is, diffusion of compounds across that alginate barrier, right? And I think in large part the demonstration with the analytical chemistry at least provided good evidence to us that we could get passive diffusion of chemicals into and out of that matrix in order to see the metabolites that are produced. And, as far as we're concerned, there don't seem to be issues associated with solubility of the S9 itself.
Okay, great. So you do have some just comments to you, well-deserved compliments on your presentation. So I'll leave those to you to respond later. So I'll move on with the questions. Some from anonymous attendee. So do we know the reliabilities of the in vitro assay results regarding estrogen activation in TOX21 that are available on the CompTox dashboard?
Do we know the reliability? I guess that would be the key term there is how are you defining reliable? I guess the question's a little broad, so I'm not sure how to answer specifically.
Okay. Okay, next one also from Anonymous. In looking at the concentration loss of the parent compounds, it appears that the corresponding concentration increase in transformation products was orders of magnitude different in units. Have you conducted mass balance analysis to identify where the other parent compound went?
I guess that's in reference to the initial evaluation work. We have not specifically conducted mass balance analysis within that data, but that data has been published, and it's something that could be evaluated if of interest.
Okay, great. So just giving you a time check, we've got 4 more minutes and there are about a lot more questions. Another anonymous questions. Attendee, have these studies been conducted using human S9?
Yeah, that's a good question. So the initial development of the method was using human S9. Obviously, we have an interest in capturing human relevant metabolism within the test system. Unfortunately, when it came to applying it directly to this specific guideline ER assay, we did observe sort of non-specific estrogenic effects of the human S9 fractions, which just meant that we couldn't use that within this one specific assay. But it's certainly something that would have to be evaluated on a case-by-case basis once this AIM method is applied to other additional assays.
Okay, next question from David Saltmiris. For the activated metabolites, are you aware of any data for in vivo half-lives?
In vivo
half-lives. So for the activated metabolites, are you aware of any data, any in vivo data for half-life?
So, well, one thing I would note is that to some degree we're strictly conducting hazard identification to identify metabolism-dependent bioactivity without specifically having knowledge of the black box of the S9 test system. And what I mean by that is we don't necessarily know post-metabolism what kind of soup that's been produced from that parent compound, right? So the abundance and identity of the metabolites within each well of that test system is largely unknown. Now, of course, based on precedent in the literature, we have a pretty good idea for a number of these compounds what those metabolites are, but it's not something that we've gone to the length to specifically, identify using mass spec to say that we've got metabolite X with this amount of abundance, and thus we can describe what its specific half-life is in vivo.
Okay, so moving on to the next one from Paul Carmichael. He begins with great talk and congrats to you on getting this work finally published. His question is, could you comment further regarding the utility of this very neat, could you comment further on the utility of this approach beyond the ER transactivation assay?
Sure, so we see applications across any assay that could be run in a 96 or 384 well format. For the most part, our interest moving forward, at least in my lab, we have a fairly heavy focus on endocrine-related assays, but that's not to say that we wouldn't see interest in applying this to some of the broader, what we call tier 1 screening efforts here within ORD, specifically with respect to assays in Josh Harrell's lab, like high-throughput transcriptomics and cell painting assays, right? So assays where you can get the broadest coverage possible in order to capture the metabolism-dependent effects, while at the same time you're capturing the activity of parent compounds, right? And then have those efforts trickle down into more specific targeted assays.
Okay, great. So I'm going to cut the questions off right now. So Chad, you have about 10 or 15 questions in there. If you can start moving your way through those. Obviously, a lot of people are really interested in this topic. So we're going to move on to the next speaker. We can get, yep, those slides teed up. That would be great. So our next speaker is David Kreiser, who is a chemist in the Predictive Toxicology and Screening Group at the National Toxicology Program at NIAHS. And so he's going to talk to us about in vitro disposition of the Tox-21 chemicals, ongoing results and next steps. So welcome, David. Are you there?
I'm here.
Okay, we're looking forward to your presentation.
Thank you for that introduction. I'd also like to start by thanking the organizers for giving me the opportunity to talk to everyone today about this Tox-21 cross-partner project, looking at the in vitro disposition of Tox-21 chemicals. Next slide, please. So currently, in vitro to in vivo extrapolation predictions rely on the nominal concentration. And so the question to ask here is, what if the nominal concentration in an assay fails to represent the cellular concentration in that assay? And the result of that is that your IVIVE prediction accuracy may be affected. Next slide. So current IVIVE prediction models are based on a steady state assumption, where you're basically making the assumption that the blood to tissue partitioning that you see in an in vivo system is equivalent to the cells to medium partitioning that you would see in an in vitro system. So on the left here, you see an example of an in vivo experiment where you would have a daily dose that you would give an animal, and then you can use a calculation to determine what a steady state concentration is. So what you're trying to do with the IVIVE method there is shown on the right, where we're trying to take a steady state concentration, for example, something like an in vitro AC50, and relate that back to an oral equivalent daily dose. Next slide. So what we're trying to get at here with this work is the fact that currently, there's not been a lot that's been looked at as far as in vitro partitioning empirically. It's been evaluated for very few chemicals and very few model systems. Thus, it's really unknown for how many chemicals and to what degree this potential differential chemical partitioning affects the accuracy of IVIVE predictions that are made across the Tox-21 chemical library. Next slide. So before we go any further, I want to bring up how this work fits into the EPA New Approach Methods work plan. So if you think about doing an in vitro study, if you don't exactly know what concentration is getting into the cells, it's really hard to say, the effect that you're seeing, what concentration caused that. So knowing exactly, how much of the chemical gets into a cell gives you a better idea of what concentration of the chemical is causing the effect you're seeing. And really the future utility of NAMs, such as in vitro assays, understanding this chemical partitioning is really important to move these things forward into a regulatory context. So this work fits into the EPA NAMs work plan under Objective 3 by helping to establish scientific confidence in NAMs and demonstrate application to regulatory decisions. Next slide. So what do we currently know about in vitro partitioning? So if we look at the figure here down at the bottom of this slide and consider a chemical that is put into an in vitro well, there's a variety of different places where that chemical can end up. First, the chemical could stay in the test medium. The chemical could also become bound to different serum constituents, such as proteins. The chemical can obviously move into the tissues or cells. The chemical could actually evaporate from the media and go into the headspace. And then finally, the chemical could get attached to the plastic or whatever your vessel walls may be. So in this work by Armitage and coworkers in 2014, they suggested that the in vitro partitioning relates strongly to the log KOW of the chemical and the concentration of the serum that you have in the medium. They also commented that the sorption of plastic played a smaller role in determining the cellular concentration. Now, there are other examples out in the literature looking at in vitro partitioning, which bring in other factors such as lipid concentrations and various other things. But for the sake of time, I'm not going to go into all of those. So next slide, please. So as we set out on this work, what is our hypothesis? So we believe that the physiochemical properties of a given chemical can be used to predict the difference between the nominal concentration, the concentration that you say you're putting into an in vitro system, versus the actual concentration that exists in the medium, and what the concentration that ends up into the actual cellular compartment. So when we're thinking about this chemical partitioning between media and cells, and in this case, we're looking at metabolically incompetent cells, there are a few things that we're interested in looking at that we believe that this is going to be dependent on. Things like the amount of serum that's in the media, the relative bonding of the chemical to serum binding proteins, log KOW of the chemical, and then, finally, is that chemical possibly going to bond to plastic? Next slide, please. So I'm going to give you an overall idea of where we're ultimately wanting to move with this project. So we hope to eventually look at approximately 200 chemicals to do a pretty decent job of covering chemical space. So these are 200 chemicals that are selected from the Tox21 chemical library. About 93% of those chemicals also exist in the ToxCast chemical library. About half of these compounds are low fraction unbound compounds, with the other half being split between moderate and highly unbound. When we consider, what charge state these compounds might exist at a pH of 7.4, about half of them are thought to be neutral, with about 30% anionic and 17% cationic. Now, just to give you an idea of how this set of chemicals, would work out in a typical in vitro assay, we looked at how they behaved in the Adagene ER assay, and about 60% of these 200 compounds were inactive in that assay. A little less than 5% were potent at less than 0.1 micromolar, with another 17.5% potent at less than 10 micromolar. Now, as an analytical chemist, when someone comes to you and says, we want to look at 200 chemicals, the first thing I thought was that's a lot of method development. But fortunately, at NTP, we currently have some sort of method available for about a fifth of these compounds that we can build off of. So there's not as much analytical method development to do. There's also, for about 3.5% of these compounds, there's a radio-labeled compound available somewhere at EPA. And the reason this is important is this gives us the ability to go back and run a different type of analysis, different analytical measurement to kind of confirm what we're going to see with the LCMS, just to give us more confidence in what we're actually seeing. So to give you some idea of what this in vitro partitioning may look like, we ran all 200 of these chemicals through that Armitage model that I showed earlier, and that model predicts that the cellular concentration will be 100-fold lower than the media concentration for 10.5% of compounds. For 14.5% of the compounds, the cellular concentration will be 3.2-fold lower than the median concentration. For about 18% of the compounds, you're going to fall within a 3.2-fold of the media concentration and the cellular concentration. The cellular concentration would be greater than 3.2-fold of the media concentration for about 36% of the compounds, and then greater than 100-fold of the media concentration for 18% of the compounds. So you can see here that, just from looking at this model prediction here, there's a pretty big range of, where that chemical might end up and how that could ultimately affect the concentration that you're seeing in your cellular fraction. Next slide. So when we first set out on our initial pilot for this study, there was a few things we wanted to be sure that we looked at and thought about and kind of got down from the start before we moved to this, huge study of 200 chemicals. So first off, we wanted to work through the sample generation and sample handling workflow and be sure we were confident in how we were generating and handling the samples, and that we could do that in an efficient way. Second on this list is probably the most important thing, and that is, are we actually getting the information that we want? And then finally, when you're talking about 200 chemicals here, there's a lot of LCMS data collection and analysis that takes place. So we want to be sure we can figure out how we can be as efficient as possible in those two aspects while we're working with a small subset of chemicals before we move to that larger set of 200. Next slide. So to start off, we decided to select 10 chemicals from that 200 chemical list to run a pilot on. And so we first narrowed down that 200 chemical list to compounds that we knew would work with LCMS. That was further kind of cut down to compounds that we knew that would ionize using electrospray ionization in positive mode, just to simplify some of the things we were trying to look at in terms of our methods of analysis. From that point, we tried to, find chemicals that had different types of functional groups, just look a little different, to try to somewhat cover, the vast chemical space that's out there. And then we also, tried to pick things. You can see the colored bars underneath the names of the compounds, which is showing the log P values of these different compounds. We tried to pick a variety of compounds that had different log P values to, again, try to cover that chemical space. Next slide. So this is the setup of our initial pilot. So we were looking at using MCF7 cells. We have 4 technical replicates of each sample that we analyze. Again, we used 10 chemicals for this pilot. We started off just looking at one concentration of 10 micromolar. We have 3 different time points of 1, 6, and 24 hours. And then there are 2 different media conditions with either 1% or 10% FBS. So the way we kind of handle the sample workflow here is cells are plated using a peristaltic dispenser into a 384 test plate that you can see there at the bottom of the slide. From there, we dispense the chemical in media using a echoacoustic dispenser. Then we, for some of the samples, we have to pull off media. And so to do that, we use a guided pipetting system from ViaFlow and move those samples onto the receiving plate. And then finally, we add acetonitrile using a solenoid micro dispenser to kind of end things up and have those samples and then put those in the freezer for later analysis. So if you see table 2 here, which shows the plate matrix, you can just look at one of the time points there and you see that you end up having to use 3 different test plates with different plating conditions in order to be able to actually measure all of these different components that we're interested in measuring. Next slide. So I'm going to go into our a little more detail about how we go about using these different plates to get to the actual value that we're interested in. So in the first test plate, you would have your cells, your media, and your chemical. And then we would, at the specific time point, add acetonitrile to do a whole well crash, which we can use for mass balance later on. Next. Then we use the same type of setup with the second test plate that again has the cells, media, and the chemical. And at the time point, we would remove the media to a receiving plate, add acetonitrile to that, and that allows us to calculate the amount of chemical that's in the media. And then what's left behind in the test plate would be the cells and the plastic that's in the well. And so we can add acetonitrile to that, and that allows us to determine the amount that's in the plastic plus the cells fraction. Next. So then in the third test plate, we now no longer have cells in there. We just have the media plus the chemical in the wells. So we would again remove the media from the test plate to a receiving plate. We can add the acetonitrile and again measure the amount that's in the media. And then what's left behind in the test plate, since there are no cells there, would just be the plastic. So we can add acetonitrile to that, and then we can determine the amount that's in the plastic. Next. Then the final thing we do is we can take the amount that's in the plastic plus the cells fraction, subtract from that the amount that's in the plastic fraction, and that allows us to determine the amount that's actually in the cells. Next. So as far as our analytical methods go, it's pretty standard and straightforward here. We use the 7-minute LC method on a C18 column. using water and acetonitrile with 0.1% formic acid in both of those as our two mobile phases. We just did a pretty straightforward, simple gradient elution. For the mass spec analysis, we used a target single ion monitoring mode. So where we just told the instrument to look for the mass to charge value of the parent ion, which helps increase our sensitivity and LOD values. Next. So how do we go about calculating all of these different concentrations and amounts in these different samples? So it's pretty straightforward for the concentrations of the chemical in the media and the whole well samples, where in order to get our final concentration, we just take our raw concentration times the two dilution factors that take place. The dilution factor where we add the acetonitrile post-incubation, and then any dilution we make prior to doing the analytical analysis. To calculate the amounts, for example, in nanograms of those media and whole well samples, we then just take that final concentration and multiply times the volume in the well to get that amount. So to calculate the concentration in the cells, again, we have to take the difference between the amount in the plastic plus the cells fraction and the amount that's in the plastic fraction, and then we divide that by the molecular weight times the volume of the cells. So for the volume of the cells, we had to search through the literature and found that, on average, MCF7 cells have a cell volume of about 2.0 picoliters per cell. So we used 10,000 cells in this 384 well setup, which gives us a cell volume of about 20 nanoliters. Next slide. So one of the things we wanted to think about is our LCMS analysis time. And that's because of, the ultimate goal was to run 200 chemicals. So I wanted to see if we could do a cassette analysis with these samples. So all of the samples are incubated individually. So you can see here at the top, single chemical incubations of, for example, 5 different chemicals, A through E. And then what we did is we did an individual sample analysis where we analyze each individual well, 1 chemical at a time on the LCMS. And then what we do is we combine 5 wells together that have 5 different chemicals in those wells to give us a combined cassette sample that we can then measure essentially 5 different chemicals from 5 different samples in a single injection, which greatly cuts down on LCMS analysis time. Next slide. So here's some data from this first run through the pilot where we're looking at the concentration in the cells compared to the concentration in the media at the 24-hour time point using the cassette analysis method. So this is the concentration in micromolars on your y-axis. And then in the blue bars, you have your cellular concentration. And then the orange bars, which are down there at the bottom that you can barely see on this figure, is the media concentration. And then above of these bars, you have 2 numbers listed. In red is the fold increase comparing this cellular concentration to the media concentration for the individual analysis, and then in the black is for the cassette analysis. So one thing, immediately we notice here is for a lot of these compounds, we definitely see a pretty big increase in the fold in terms of fold change from comparing the cellular fraction to the media fraction. And then second, we see pretty similar results between the individual and cassette analysis, which gives us some confidence that this cassette analysis could work. Next slide. Now, the problem we ran into here is when we go to compare the plastic versus the plastic plus the cells fraction versus the plastic fraction. And we see pretty small differences, anywhere from a half of a nanogram up to a little over 10 nanograms. And so when we think about the dilutions that are taking place and trying to do this cassette analysis, which is going to further dilute samples, we're really running into issues in terms of sensitivity and limit of detection. So that's creating a problem, allowing us to accurately measure what that cellular concentration is. Next slide. So from our first run through the pilot, the things we learned was that the cassette analysis for the analytical measurements looks like it produces pretty similar results to the individual analysis. So this would give a major reduction in LCMS analysis runtime going from 282 days, 24 hours a day, to 56 days, 24 hours a day, which is actually something reasonable to obtain. And then again, we ran into this big challenge of the small differences in the amount of chemical observed in the cells versus the amount that's bound to the plastic. Next slide. So we then decided to move into a second run through a pilot. And this time we decided to move from the 384 well format to the 96 well format. What this does is it gives us a greater number of cells in each well per surface area. We also decided to add in a wash step after removing media from the test plate so that we could be sure to remove any residual chemical that's not actually in the cells or bound to the plastic. And then finally, the last change from the first pilot is that we now only use a single media composition with 10% FBS. There were some issues with the cells and the way they looked at 1% FBS, so we just decided to move forward with the 10% FBS media condition. Next slide. So in the initial LCMS analysis of these pilot 2.0 incubation samples, I noticed some pretty big issues with LCMS drift over the 2 or 3 day run times. And so I went back and reanalyzed the samples from those incubations, but this time used a data normalization method to account for this drift that we see in different changes in the LCMS instrument over that runtime. And to do this, I injected a standard every 10 injections that can then be used to normalize the signal intensity across the run, which really helped with being able to deal with these drift issues. Next slide. So when it comes to calculating the concentrations and the amounts for the 96 well format, everything is essentially the same until you get down to the concentration in the cells calculation, where now what's changed is our volume of the cells. So now instead of using 10,000 cells, we're using about 50,000 cells per well. So we now have an increased volume of cells up to 102.2 nanoliters, which we have to use in that calculation to calculate our cellular concentration. Next slide. So this slide shows some data that compares the media concentration versus the whole well concentration at 24 hours. And so in the blue bars, you have the media concentration, and the orange bars is the whole well crash concentration. And so the take home really from this slide is to show you that for almost all of these chemicals, what we see is we see a higher concentration measured in the whole well crash compared to the media, which is exactly what we would expect, because obviously there are more compartments that the chemical can move into besides the media. So we'd expect that whole well concentration to be higher. Next slide.
David, 2 minutes, please.
Okay, so when we looked at the concentration in the cells versus the media at 24 hours, we again see for a few compounds a big increase for a couple of these compounds in the cellular concentration compared to the media concentration. But for a lot of the compounds, we were not able to see that difference. So we wanted to figure out why was this happening. Next slide. So what we realized is we started to think about our actual in vitro system here. And so if we look at these plots here, which are showing the plastic versus the cells amount on the y-axis, and then the amount that's in the plastic and the x-axis, you can see on the left plot where we assume that all surface area is available for plastic bonding, that the mass in the cells is at or near zero. And that's why we're only able to measure that cellular concentration for a couple compounds. But on the right, if we consider that only the walls are available for plastic bonding and not the bottom, we're now able to get an actual measurement of mass in our cells, which allows us to calculate that cellular concentration. Next slide. So this is just a quick comparison to show that our measured concentrations match up pretty well with the Armitage model. Next slide. So we did have an issue with some mass balances with the cassette analysis method, but since this time, I figured out the issue here, and it's just a matter of the types of seals we were using on our plates. So I think I have this sorted out. Next slide, if I can wrap up. So we do see that nominal concentration doesn't equal cellular concentration. Unless we consider that the surface area on the bottom is not available for bonding, we have those near zero cell concentrations. But making our assumption that the walls are only available for bonding helps fix that. Armitage model gives a pretty good estimate of our cellular concentration, and I think I have this mass balance issue figured out. Next slide. So we are moving to a new pilot where we've added in 10 more chemicals, and we're increasing to 3 concentrations just to allow us to better able to, study this and see the trends that are going on. Next slide. Here's those additional 10 chemicals, same type of thing, trying to, continue to cover the chemical space better. And then next slide. So we have the analytical methods ready for all of these 20 compounds. Incubations were completed in February before we were kicked out of the lab for COVID. And so we'll probably get the analysis done on these the beginning of 2021 and have some more data on this project. Next slide. And just to finish, I thank all my coworkers that have been a great help in helping this project move forward. And I'll take any questions.
All right, thanks, David. You have quite a few questions, and we won't get to all of them in 4 minutes. So a couple of them are quite technical, so I'm going to jump down to something a little bit more big picture. So from Rocky Goldsmith, for the chemistries you have looked at, are there specific physical chemical parameters or molecular descriptors or fingerprints that suggest one could identify chemicals in well-behaved or poorly behaved chemistries in the in vitro context? Molecular features or properties that could suggest assay amenability and flags?
Yeah, we haven't dug down into things that far. We're really trying to make sure we, nail down the, analytical workflow first before we, try to really start digging down and looking at all of those trends to actually, what properties matter and how chemicals partition. But that is something that we're going to dig into with this next pilot, and hope to actually publish that larger pilot study to kind of go into all those details.
Okay, great, thanks. Okay, and question from Martin Phillips. Have you considered using NMR to quantitate using stable labeled compounds versus radio labeled ones? Could this increase the number of compounds you can ground truth?
That's not something we have considered at this point, but that is a good point.
And a question from Anonymous: why would there not be binding at the bottom of the wells?
Yeah, so these MCF7 cells are pretty confluent across the bottom of the wells, and it's almost 100% covered. I don't know the exact percentage, but it's very close to 100%. So you're really that chemical is not going to move through those cells and into the plastic there. So we feel pretty comfortable in making that assumption.
Okay, we've got a couple more minutes. I'll keep going through the list. Are the NTP methods validated in GLP quality?
They are not to that extent, yeah.
Okay, next question. Are you counting via flow cytometry the number of cells? So that may be for the previous speaker. What is the effect of xenobiotic efflux pumps, which can be upregulated?
Yeah, so we chose to initially start off with the MCF7 cells to kind of cut out this issue with, potential metabolism, but, there are some different things that can still happen in these MCF7 cells. And I think that's part of our reason for, kind of moving to a larger pilot with more chemicals and more concentrations to be able to make sure we, fully understand everything that's going on. We just don't have enough data at this point to, make any comment on that.
Okay, so last one, unless it's super quick. Are you counting via flow cytometry the number of cells to calculate the total volume of cells for determining chemical concentration in cells?
I'm not exactly sure how Josh Harold did the actual cell incubations, how he calculated those. So we would have to reach out to him for how he's calculating those number of cells.
Okay, so we can respond to that one in writing via email on another day, I guess.
Okay.
And this will be definitely the last one. Does your work largely validate the Armitage model? Do you expect to see departures from Armitage, and why?
I mean, so far, it looks like there's pretty good agreement. We do think that there are, and there's other things out in the literature that suggest there are other factors that's not included in the Armitage model. So that's what we're really trying to do here is, to see is it possible to, develop a, more involved model that considers, more physiochemical properties and different things like that to, improve upon these predictions.
Okay, great. Thanks so much, David, for a fantastic presentation and helping out with the questions. So you do have a number of questions in the Q&A box. If you need help responding to them, ICF is in the background to help out if you need them.
Okay.
Okay, so our next presentation, we're gonna change topics a bit. So we're gonna have a presentation from Clemens Whittier from the Joint Research Center on European Commission in ISPRA, Italy, on OECD harmonized templates to report NAM results in regulatory environments. So this will be the first presentation in a series, followed up with 3 case studies looking at regulatory and risk assessment application of NAMs. So, so we're going from more detailed laboratory work into practical applications now. And we appreciate the Clemens for joining us today. And just remind all the participants as you ask questions, because the question box is definitely getting more and more active, that you please put the name of the speaker in front of your question, because some of them are getting stacked within each other. So the questions from Chad and David are getting a little bit mixed up. So please put the speaker name in front of the question. It'll help all of us respond to you in kind. So with that, I'll turn it over to Clemens.
Okay, thank you for this introduction, Anna. And buonasera to all of you from Italy. I would like to thank the organizer for having me here in this conference. It's really great to get the opportunity to present one of the international angles and also the data sharing aspect of the NAM story here today. So let's get started. Next slide, please. What can you expect from my presentation today? I will briefly introduce the concept behind what we call the OECD harmonized templates, or OHTs. Then I will zoom into one of the templates, to one template used for reporting mechanistic information, which we call the OHT 201. Then I will talk about what we here at the JRC call the triangle of chemical safety and what role the OHT 201 plays in it. And finally, I will show you how OHT 201 looks like in real life by giving you a sneak preview into our upcoming endocrine active substances information system. Next slide, please. So let's get started with an overview of what what OECD harmonized templates are and why they are so essential for global data exchange. This is a very simplified diagram of what the world looks like as long as there is no agreement on data formats. On the left-hand side, you see the data producers. On the right-hand side, the data consumers, the authorities that are supposed to trust the data they receive. Don't get hung up on the exact flags I used here. There are just small random sample of countries that are in the business of chemical regulation. In the middle, you see a jungle of different fancy formats, and the arrows crisscrossing the slide show how incoherent and even chaotic the data landscape can be. Data exchange between authorities or between data producers and among them is next to impossible, and there is obviously lots of double work and double effort involved. So this is not something we should aim for. Next slide, please. I think you agree that this picture looks much, much better. Instead of lots of different formats, there is one single internationally agreed template which all data producers use and which all authorities accept. Again, the diagram is very simple and a depiction of a very complex real-world situation. But still, the principle is clear. Once authorities start accepting the same reporting format, transnational data acceptance can start. And if the template in the middle deals with mechanistic information, NAM-derived data suddenly become much more visible and available across countries. The arrows at the right-hand side of the slide show you how happy authorities now start to share data and start acknowledging data from each other. Now, I guess that looks much better than the first slide. Next slide, please. Sorry for getting so basic, but I think it's useful to point out what definitions for apical and mechanistic knowledge we use in this talk. So apical knowledge is obviously the knowledge about traditional directly measured whole organism outcomes of exposure in vivo tests, generally death, reproductive failure, and so on and so on. So one in vivo test tells us whether an adverse outcome has been observed or not. While mechanistic knowledge is the knowledge about the sequence of events leading from exposure of to an effective dose of a chemical to the production of a specific biological response in the target organ. In most cases, in non-in vivo tests. So while apical knowledge is measured mostly in one in vivo test, mechanistic knowledge can show us a series of tests, mainly non-animal, where how an adverse outcome is likely to manifest itself or not. Next slide, please. This slide shows a brief history of the OECD harmonized templates. Back in 2002, people started to think that global harmonization of the way chemical test result reporting should happen, that harmonization would actually be a good idea. Templates and even XML tags were mentioned back then. 2 years later, in 2004, the OECD then actually established the OECD Expert Group on Harmonized Templates, which delivered a first batch of 86 templates only 2 years later. And I have a strong recollection of the many exciting and not always conflict-free discussions we had between industry and authorities when we discussed the structure of these 86 templates down to field level and even the exact wording of the help texts. After that, and until today and beyond, much more of such templates were created, agreed upon, and put into use, and that's still happening. But all these templates are apical, or were apical, and around 2012, we started to think, hold on, how do we report non-apical data? How do we make sure that mechanistic data get much more visible and get into the mainstream of regulatory thinking? So we started to draft a mechanistic template. That template now got the name OHD 201 at the OECD, and it took us quite some time to get it right and to get it past OECD approval and agreement. Finally, in 2017, OHD 201 became official. However, the rapid evolvement of NAMs and the kind of data they produced, and also the necessity to better link the mechanistic data reporting to the adverse outcome pathway framework, made us come up with a revamped version of the template, which, as we speak, is in the final phase of adoption. We expect the revised template to be adopted at the OECD in November. Next slide, please. So to summarize, OHTs are only descriptions, data descriptions, and are not yet an ICT application. OHTs can be implemented by anyone in their local IT environments. However, there is already a very popular OHT implementation around, and that's Euclid in its current version, Euclid 6. The Euclid development is managed by the OECD, and Euclid is free and can be installed in any ICT environment. So you might wonder, that's nice, but where are these OHTs and Euclid actually really used these days? Next slide, please. Well, the answer is in more and more countries. The slide shows the current situation, and you see that my flag selection on the first slide was actually not random after all. Australia, Canada, New Zealand, Switzerland, the US, and the European Union do actually use the OHTs already now in their environments. I don't want to go into the details here, but the US already uses Euclid and OHTs in 3 topical areas. I guess you know these areas better than I do, but these are things where the OHTs are already used in the US. And that is definitely not the end of it. Japan is already very interested, and some other countries as well. So this is really now getting global reach. Next slide, please. So what you heard until now was valid for all of the more than now 100 OHTs. Let's now zoom into the, as we call it, odd one out, the mechanistic template, if you want. Next slide, please. So again, why do we need a mechanistic template? Why did we invest so much time and effort to come up with OHT 201? Obviously, all other OHTs are apical, as we said before. Apical means referring to one single endpoint of regulatory concern, like fish toxicity, bird toxicity, if you go into ecotoxicity, or skin sensitization, carcinogenicity, acute toxicity when you go to human health. But mechanistic data are not intrinsically linked to one single apical endpoint, and that's very important. They can be used to underpin mechanistic explanations of toxicity across and beyond apical endpoints. Please note the little diagram at the right-hand side. One single instance of OHT 201 can be relevant for more than one apical endpoint. And this is the reason why mechanistic information should normally not be reported in an apical OHT, because that way it would be inseparably locked into that one endpoint and would not be, and it would not be possible to easily reuse it for the explanation of mechanism beyond this one endpoint. Next slide, please. Okay, so let's have a closer look inside the OHT 201. The first thing that needs to happen, obviously, is that you have a chemical and you apply a test method, a NAM to it. Out of this NAM, you get one measurement or two measurements or several measurements. Out of that, and that is the intellectual contribution of the individual who fills in the OHT 201 template, you arrive at one single subjective conclusion. Either the chemical you observed now did show the mechanistic effect you describe, or it did not. So several objective measurements lead to one subjective yes/no conclusion. Obviously, again, it's not as simple in the real world. There aren't always clear yes/no answers, but and obviously these gray zone answers can also reported in OHT 201. But that's the basic principle. The big box in the middle shows that the effect you observed or did not observe is actually not just randomly named, but tagged with a triplet of ontology terms. Each mechanistic effect is identified by a process object action triplet, and we will come to that later. Next slide, please. OHT 201 would be of very limited use if it locked you into a specific technology or class of technology. Mechanistic effects can, as you all know, be measured or determined with all kinds of technologies, in vitro, in silico, in chemico, omics, and you name them. So the evidence you present in the OHT 201 can be derived from any of these technologies. That's very important, because else we would not cover all possible data that could be an important piece of the puzzle. OHT 201 is not an in vitro template, but it's the intermediate effect or the mechanistic information template. So it's not just for in vitro information. Next template, please. Next slide, please. Just a quick remark about the process object action triplet. For tagging or naming the intermediate effects we report with OHT 201. We use the same ontology terms for tagging intermediate effects in OHT 201 that we also use for naming key events in the adverse outcome pathway framework and then physically in the AOP knowledge base. So an OHT 201 intermediate effect is more or less equivalent to an AOP key event. You will appreciate the beauty of this when I show you the triangle of chemical safety a bit later. Next slide, please. Here you see a real-life example of this tagging taken from the adverse outcome pathway protein alkylation leading to liver fibrosis. You can read for yourself, but each of the key events is here tagged with a process object action triplet plus a context tag, which is additional information. So let's have a look at the, for example, the stellate cell activation key event. It has a process, that's hepatic stellate cell activation. It has an object, obviously hepatic stem cell. It has an action, which is increase. So you see how it works. You have the process, which is activation. You have the object on which this process works, and the action is either increase, decrease, or downregulate, upregulate. In this case, it's an increased mechanism. And if you measure the stellate cell activation of a specific chemical with your preferred test method, whatever that is, you want to report the results using OHD201, and you want to tag the OHD201 with the exact same ontology terms, then a connection starts to emerge. And I think you see where this is going. Next slide, please. You remember that a couple of minutes ago, I said that OHD201 is technology independent. Well, in real life, we obviously want to increase the usefulness of the template for the most popular, or at least the most data-intensive classes of NAMs. So we introduced very well thought through structured reporting fields geared towards in vitro methods, both test guideline methods and non-test guideline methods. And I'm talking about OECD gets test guidelines here. All other data, or all data derived from other technologies, obviously can, as said before, obviously also be reported, but then more relying on free text fields, attachments, or links to third-party resources. But the same still applies. You report one subjective conclusion, effect yes, effect no, using OHD201. Next slide, please. Speaking of OECD test guidelines, most of you will be familiar with them. These are not, these on this slide are the test guidelines that are already fully supported in the upcoming version of OHD201. And if you report data that were produced following one of these guidelines, many fields in the template are already pre-filled. If you didn't follow a test guideline, absolutely no problem. OHD201, as I said, is technology independent. You just have a little more work when filling the template in. And in the coming years, much more guideline-sensitive support will be implemented in the template. So the list you see here on this slide will get much, much longer. Next slide, please. So now let's fit all the pieces together and talk about what we like to call the triangle of chemical safety. Next slide. The corners or angles of this triangle are the stressor, the test method, in vitro or in vivo, and the adverse outcome, or the key event of an adverse outcome, to be more precise. Next slide. So what happens is that a chemical undergoes a test, and the test shows that the chemical either triggers a certain key event or it doesn't. In some cases, the result might be inconclusive, as we said before. The world isn't always black and white or green and red in that case. But remember, it's your subjective conclusion that you report. Yes or no, or not quite conclusive. Anyhow, whatever your conclusion, you stick it together with the objective measurement data into an OHD201, and voila, the triangle is complete. Next slide, please. And this triangle gets even more complete when you tag all three angles of the triangle with the same ontology terms. Because wouldn't that be the holy grail? I mean, key events, test methods used to measure them, and the test results themselves tagged with the very same ontology terms. It sounds still like a bit of science fiction, but wouldn't it get much easier to find test methods that are suitable for specific key events, or the other way around, find key events for which a specific test method can be used, and on top find real-life measurements with real-life chemicals that align themselves with key events? That is what we should try to achieve in the coming years, and we at the JSC already are working on that. Next slide, please. But obviously, let's not get carried away by this science fiction talk. Let's go back to the real world and find out how our good old OHD201 behaves in the real world. Next slide, please. In a few weeks, the JRC will publish the Endocrine Active Substances System, acronym is EASIS. And at the day of its publication, and we hope it's November, we are still fighting some administrative obstacles here, but hopefully in November, EASIS will feature data from 629 substances derived from more than 10,000 studies, 7,000 health effects, 2,500 effects on biotic systems, and already 550 intermediate effects. So that means once EASIS gets published, we will already have 550 OHT 201s filled in EASIS, and everyone can browse through them. The right-hand side of the slide shows a diagram of how we manage and populate EASIS. So you see that we have third-party service providers who have their own Euclid installations. They go through the literature, they browse through the scientific literature, extract data, code the data into Euclid. If it's mechanistic, they code it into OHT 201. They send the data to us. We include that in what we call the JRC EASIS workbench. We manipulate the data a little bit, clean them up, and ultimately populate, copy them into the public website. So the next, thank you. The next few slides show real-life screenshots from EASIS. This here is the chemical overview. You see we have 629 chemicals, and the page shows 2 of them. Next slide, please. Now we zoom into one of the chemicals, and the left-hand menu shows the OHTs that have already been populated for this chemical, 1 with general information, 4 environmental information, 2 about health effects, and then 12 for intermediate effects, and that is obviously OHT 201. Next slide, please. Now we zoom into one of these OHT 201s, and the following slides will mimic scrolling down a small part of the template. I will not go into the details. I invite you to visit EASIS once it's public, and you will see the fields correspond. The fields in EASIS correspond exactly to the OHT 201 reporting paradigm: effect naming, test method description, several objective measurements leading to one subjective conclusion. One caveat: these screenshots show, shall we call it, the old version of OHT 201, because the new one is not yet publicly available. So now
3 minutes.
Yeah, okay. Just quickly flick through the next screenshots. I just edit them for completeness. Okay, next one. Next one. Next one. Next one. Okay, so the take-home messages is OHT 201 facilitates reporting of NAM study results in an internationally agreed format. OHT 201 supports the chemical angle of the stressor method, the AOP triangle. OHT 201 is available in a free ASI application, Euclid 6. OHT 201 supports all classes of NAMs, but especially in vitro methods. And OHT 201 reports per chemical and intermediate effect 1 or more objective measurements and 1 subjective conclusion. Next slide, please. Yeah, we were also asked to include a slide about how our work fits into the EPA NAM work plan. Here we go. OHT 201 is explicitly mentioned in the work plan. So it's already acknowledged in the work plan, and we are standing by to help you if you want to expand the importance of OHT 201 in your work plan. Just let us know. Next slide, please. Few links if you want to know more about the stuff I was talking about. And last slide. Yeah, that's it. Thank you very much for your attention. Let me acknowledge all the people that were involved in the work from OECD, ECHA, and from the JRC. Sorry for not including a slide featuring the name. Sorry, I forgot that. So really sorry. But this was a huge team effort and showed how important things can actually be achieved in teleconferences instead of face-to-face meetings. And that was maybe another important lesson we learned in the process. So that's it from my side, and I'm looking forward to your questions.
Okay, great. So we do have some questions, and they're starting to come in quickly. A couple of them will be easy to do in written response, so I'll start with some of the bigger picture ones. So from Anonymous, this approach feels very human-centric. How do you envisioning applying to ecological applications for AOP? Should species domain be included in the OHT?
Well, the OHT 201 is neither human-centric nor ecocentric. It's just if you measure something in vitro or in silico, or you can report it in OHT 201. So that really, let's say that the domain of applicability is not limited to the human or the ecotox area, because as I said, you should not try to inseparably link your in vitro measurement to an adverse outcome, and thereby you cannot, let's say, the link to either eco or human health is, I don't see it.
Okay. Next one from, let's see, is there any plan to either amend the OECD TG guidance with the standardized OHT 201 templates, or have them co-located with the TG on the OECD website, TG being test guideline?
Yes. The next step that, or let's say the next big chunk of work that we will have to go through is come up with guidance, and to exactly show people how, when they want to follow a certain guidance, how they have to use OHT 201 or not OHT 201, because there might be cases where a test guideline is better served by not using OHT 201. But the general principle is, if you have a NAM method, you use OHT 201, and the guidance we will come up with.
Okay. Next one from Carol Marchant. Is the ontology linking to OHT 201 already in place, or is this work to be done?
This is, we reused ontologies. We did not come up with new ontologies, so we tried to reuse existing ontologies, and the selection of the ontologies we use, and also the selection of the ontology terms we suggest, is already done. Yes.
Okay, next one from Barbara Burke. How is the GIVIMP, so the good in vitro practice guidance from OECD, included in the OHT process?
GIVIMP and the OHTs are inseparable. So my colleague Sandra Kuke, who was responsible for here at the JSC coming up with GIVIMP, and I, we were very close, working very closely together to make sure that if you want to report data under the GIVIMP paradigm, that you would use OHT 201. Yes, that's a very close relationship. Yeah.
Okay, great. Next one from Yad Buller, PMR Canada. Is the OHT 201 also designed for in silico approaches such as read across and QSAR?
Yes, yeah. As I said in my presentation, the OHT 201 is technology independent. However, knowing that there are many people who want to report in vitro data, we already foresaw many structured fields for in vitro test methods. But if you have an in silico approach or an in silico observation or measurement or calculation, you can obviously report it in OHT 201, because the important part of the OHT 201 is your subjective conclusion, and that is totally independent of the technology that you use.
Okay, so next one from Anne Millen from OECD Secretariat. Great talk, Clemens. How does OHT 201 match with the OECD guidance document 211 on the description of non-standard guidelines? And maybe you can say what 211 is for those of participants who aren't as close to the OECD.
There is often a confusion because they are unluckily named with a very similar number. So OHT 201 is the description of how to report in vitro or NAM data, while guidance document 211 is the description how you describe the method description, what you have to do when you execute the method. And this is actually referring to this triangle I showed. The 211 parrot 211 guidance document should refer to how you describe a method in the if you remember my triangle, in the right upper hand angle of the triangle, while OHD 21 is in the left upper hand angle of the triangle. So both have their right to exist in the triangle of chemical safety.
Okay, great. So we've hit exactly 11. You have a couple of questions in there that respond to one overlaps with a different one, and then new question. And there is a question for OPP and OPPT EPA speakers that we'll respond to in writing later in the day. So we've hit 11 o'clock Eastern time, and it is time for our 15-minute break. So if, for those of you on the US East Coast, we'll meet back together 11:15. Everyone else, check your watches, and we'll see you back here in 15 minutes for 3 case studies, really providing practical application of NAMs in the risk assessment space. So we'll see you back in a few minutes. Okay, everyone, welcome back. Hopefully you got some coffee or water or stretched your legs a touch or answered some emails. I know I certainly did all of those things except the coffee part. Moving on to the case study. So I introduce a colleague of mine, senior toxicologist at EPA's Office of Pesticide Programs, Monique Perrin, who's going to talk about some really exciting collaborative work the Pesticide Office is doing with the Office of Research and Development in collaboration with EFSA and a number of other organizations, and some information we presented to a federal advisory committee last month, actually. So a lot of this is quite hot off the presses. So passing it over to Monique.
Okay, thank you very much, Hannah. So today I'll be—
Monique, we can't hear you.
Oh, you can't hear me? Hello? Can you hear me now?
Oh.
Hello?
Oh,
Monique,
actually,
we can hear you.
we can hear you. It was me. I didn't have my headphones in.
Oh, okay.
Sorry.
I was going to say that I have something wrong with my computer. Well, thank you. And I have the pleasure of presenting the first case study today, which is about the integration of NAM data for evaluating potential developmental neurotoxicity. Next slide, please. And this is the typical disclaimer that you'll see where this presentation has been reviewed and approved in accordance with EPA policy, and any mention of trade names, products, or services does not imply an endorsement by the US government or the US EPA. Next slide, please. So pesticides are regulated in the United States by EPA under the Federal Insecticide, Fungicide, and Rodenticide Act, FIFRA, and the Federal Food, Drug, and Cosmetics Act, FFDCA. Both of these were also amended in 1996 by the Food Quality Protection Act. Under the Code of Federal Regulations, 40 CFR Part 158, you can find an outline of the data requirements for pesticides, and this depends on the expected use pattern for that chemical. So is it a food or a non-food use can drive which data is required, as well as the expected routes and duration of exposure. Those toxicological studies, though, provide the EPA with information on a wide range of adverse effects, routes of exposure, duration, species, and life stages. And as such, pesticide registration requires a large number of studies that utilize a large number of animals and resources. Next slide, please. So I'm going to be talking about developmental neurotoxicity, and when that refers to any adverse effect of exposure to a toxic substance on the normal development of the nervous system, structures, and/or functions. And a DNT study is performed in accordance with EPA or OECD test guidelines. And under Part 158, it's conditionally required using a weight of evidence approach that considers these 4 considerations that are listed here: whether the pesticide causes treatment-related neurological effects in adult animals, whether it causes treatment-related neurological effects in developing animals following pre- and/or postnatal exposure, the pesticide elicits a causative association between exposures and adverse neurological effects in human epidemiological studies, and if the pesticide evokes a mechanism that is associated with adverse effects on the development of the nervous system. Next slide, please. So the design of this study is designed to develop data on potential function and morphological hazards to the nervous system that could arise in offspring from exposure of maternal animals. So the pregnant animals are administered the test substance during gestation and lactation. The offspring are then randomly selected for a neurotox evaluation, which includes assessment of several parameters, which includes motor activity, learning and memory, and neuropathological exams. Next slide, please. We do prefer an information-based approach to determine if a standard guideline study should be required or if an enhanced study or an alternative study should be considered in lieu of the regular guidelines study. And in some cases, this approach may also provide us with support for a study waiver. So as a risk-based program, the Office of Pesticide Programs considers both the hazard and the exposure information when evaluating the need for a DNT study. And if an alternative study is agreed upon, we will ask registrants to provide testing protocols and supporting rationale prior to the study initiation. Next slide, please. However, over time, several challenges and limitations have been identified for the DNT guideline study. One is the reliable detection, measurement, and interpretation of DNT effects depends on the appropriate study design and conduct that adequately controls for factors such as variability, impact of systemic toxicity in maternal animals and offspring, the experimental procedures, and the environmental conditions. So as a result, a weight of evidence approach is critical to assessing the DNT potential in a guideline study. The study also infers the DNT effects are based on apical endpoints with little or possibly no information on the underlying processes that may be responsible for those. Also, data interpretation is hampered by limitations such as high variability, low precision, and it's a very resource intensive study from a financial, labor, and animal use perspective. And furthermore, the data interpretation can be challenging for when we see only isolated findings where one endpoint is not substantiated by other findings in the study. And lastly, there are challenges with correlating those effects in the animal model to the complex neurological effects in humans. Next slide, please. So at the Office of Pesticide Programs, we've reviewed approximately 100 DNT studies. And to date, about 20 of those have been used to set points of departure for risk assessment. All of those were based off of offspring effects without any corresponding maternal effects. However, in many of these cases, the offspring effects would have also been picked up in another study, such as the reproduction toxicity study, so the pup mortality and the pup weight and things like that. And also for chemicals that we have a known neurotoxic mode of action, like the organophosphates, the N-methylcarbamates, and the pyrethroids, we do have studies available for 32 of those chemicals, and none of the DNT studies provided the most sensitive endpoint for human health risk assessment. So overall, to date, we're not seeing a large impact of the DNT guidelines study on pesticide human health risk assessments. Next slide, please. And we really started to shift our testing from the DNT guideline study to more targeted testing, particularly when there's a commonly accepted mode of action. So for the OPs and the NMCs, we require comparative cholinesterase assays to evaluate acetylcholinesterase inhibition across life stages. For the pyrethroids, we've utilized in vitro studies and PBPK models, the physiologically based pharmacokinetic models, to evaluate life stage susceptibility. And then we've also, if we have a concern with respect to thyroid toxicants, we have a comparative thyroid assay that we would determine if we think that that study should be required instead. Next slide, please. So the inclusion of new approach methodologies that take advantage of newer technologies is the logical next step for OPP in our efforts to implement more human relevant and efficient approaches. And our colleagues at the Office of Research and Development have been part of an international effort to develop a strategy to address several of the issues that I mentioned that have been identified for the guideline DNT study. And this strategy is focused on developing in vitro assays that assess the critical processes to the development of the nervous system, And these efforts have resulted in a battery of in vitro tests developed by ORD, as well as investigators funded by the European Food Safety Authority, or EFSA. And again, that these focus on processes that are critical to the development of the nervous system. Next slide, please. So currently, the assays developed by ORD use two primary technology platforms. The first is high content imaging, or HCI assays of neural cells, which evaluates 21 assay endpoints to understand processes, including cell proliferation, apoptosis, neurite outgrowth, and synaptogenesis. The other is microelectrode arrays, or MEAs, to evaluate the neuronal network formation. This platform includes 36 assay endpoints that indicate general neuronal activity, bursting, and connectivity. In addition, both of these platforms also incorporate a measure of viability. And you'll notice on this slide, there's also the zebrafish on the further right-hand side. And ORD has been actively working on efforts related to using the zebrafish assays for DNT evaluation, assessment of behavioral effects during nervous system development in the zebrafish embryos has been proposed as a complementary model for the DNT, given the conservation of neurodevelopment and functional processes across species. And it also can complement in vitro testing systems by having the advantage of looking at changes in the intact animal model. But the work on the zebrafish is ongoing by a consortium of laboratories, including ORD, to develop and define a unified experimental protocol at the OECD level. So as a result, this assay is not yet mature enough for incorporating into the DNT NAMs being considered right now, might be in the future. And so the case study today I will be presenting will be focusing on the HCI and MEA data. Next slide, please. So overall, the DNT NAM battery developed by ORD is comprised of 57 assay endpoints in human and rat neural cells that evaluate the majority, but not all, of the critical processes of neurodevelopment. As you can see here by the blue is indicating the different assay for each of the different critical processes. So things like differentiation and migration are not covered by the HCI and MEA assays. Next slide, please. So the reproducibility of the MEA and the HCI assays can be evaluated or was evaluated to understand the dispersion of the distribution of raw results for the control DMSO treated wells. This coefficient of variation, or the CV, of the vehicle control wells in these assays are generally less than 20% and suggest that the assays are reproducible. So the MEA had about an average CV of about 16% and the HCI of approximately 9%. For the MEA, we were also able to evaluate chemicals for repeat screening. So 21 test chemicals were screened across more than one experimental block using more than one sample of the chemical. So this gives us an understanding of the hit call and potency reproducibility for the test chemicals. So you can see there were 3 response groups. We had 10 out of 21 chemicals had a strong positive or negative response. 5 out of the 21 had what was considered equivocal, and 6 out of 21 had a mixed response. So altogether, from a qualitative perspective, the replicability of the overall test, overall chemical hit call was about 71%. And in terms of quantitative concordance, for 9 out of 10 of the chemicals with a strong response, we were able to show that the average standard deviation was approximately within, was less than 0.5 log micromolar of the, for the log AC50 values. We couldn't do the same type of analysis for the HCI assays because there were no chemicals that were screened more than once in that assay. Next slide, please. So in terms of assay performance was evaluated with assay controls that are known to cause effects on neural network formation and function for the MEA and for the HCI endpoints that inhibit the particular neurodevelopmental processes we're looking for in vitro in those assays. So for the MEA, so here we have a heat map with the different controls. The top 5, I know you can't really see the names, but the top 5 are the positive controls, and the bottom row is for acetaminophen, which is a negative. And as you can see, the acetaminophen appears to be an optimal negative control with no positive hit calls in any of the assay endpoints. And all of the other assay performance controls decrease general activity, bursting activity, and network connectivity measures rather than increasing any of those activity types. And overall, we see that we did do a Z prime calculation, and we did find those to range from about 0.55 to 0.8, suggesting that these controls demonstrate reproducible, robust results for the MEA. Next slide, please. So for the HCI assays, to look at the performance controls was a little bit different because they were screened as on-plate assay controls at a particular concentration, so we didn't have multiple concentrations there. And so if something in this heat map, if you see a gray square, that was because that assay control is not used as a, for that particular assay. But what we did see was that the positive control for the proliferation was, clearly produced effects, but had a slightly lower magnitude that led to a lower signal to noise and Z prime value. But overall, otherwise, we saw that the positive controls were, moderate to optimal for the HCI assays. So altogether, what we're seeing from these analyses is that for both the HCI and the MEA, the assays appear to be robust and reproducible for fit for purpose application to identify putative DNT bioactivity with reasonable assay performance controls established for them. Next slide, please. So then we wanted to consider what would be a good case study, and this I believe was decided years ago, but for the organophosphate pesticides were selected as a for the case study. These chemicals are currently undergoing registration review, which is a process by EPA to review each registered pesticide at least every 15 years. So we currently have 22 OP compounds undergoing registration review, and they were primarily selected given the large amount of data available for these chemicals and the widespread acceptance of their mode of action, their neurotoxic mode of action. So they were a great group for us to use as a case study for the development of this battery. Next slide, please. And for those of you who might not be familiar, the OPs are a class of insecticides with numerous uses, including agricultural applications and mosquito control. In 1999, we established them as a common mechanism group, that they all share the ability to bind and phosphorylate acetylcholinesterase in the nervous system, and that leads to an accumulation of acetylcholine and ultimately neurotoxicity. So the acetylcholinesterase inhibition is used as the basis for the OP risk assessments at the Office of Pesticide Programs. And just a small note that, excuse me, that some of the OPs do require activation to their auxon metabolite, which is considered the active moiety, and some of those were included in this case study. Next slide, please. And another reason why this case study is really important to the Office of Pesticide Programs is because there have been newer lines of research on OPs that have raised uncertainty with respect to potential developmental neurotoxic effects. And the interpretation has been challenging due to some conflicting results. And we also don't know what the mode of action is for these neurodevelopmental outcomes. We did hold a series of scientific advisory panel meetings in recent years to look at epidemiological data to inform the OP compound chlorpyrifos, and also it's the relevance of that data for other OPs. But the reports have provided us with several recommendations for additional research, and also at times conflicting advice for how to consider or not consider the epidemiological data. So the data from in vitro assays may be considered as part of an overall weight of evidence, or WOE, to determine the DNT potential for individual OPs, and this could be used in the future for informing our Food Quality Protection Act safety factors that are in place for concerns for children and infants. Next slide, please. So here we have the results of the HCI assays. And in terms of potency, nearly all of the log 10 AC50 values range from approximately 0 to 2 or 1 to 100 micromolar, with only a couple exceptions. And what you see here is different clustering. So cluster 1 are OP samples where you saw really no effects or disparate patterns of response in 1 to 3 assays. The second one, you have OP samples with effects on 5 or more assay endpoints. Cluster 3, you start to see the OP samples have effects on all of the assay activity types except for neurite outgrowth initiation. And cluster 4, you just see a more widespread effect across the activity types.
Monique, 3 minutes.
Oh, next slide, please. So for the MEA, we had similar potency, and here we saw more of like 3 clusters where either the samples had less than 3 assay endpoints that were positive. You had a couple where you saw discordant patterns, and then the larger group where you really saw general activity bursting and network connectivity were all decreased, and that's that larger group at the top. Next slide, please. I might need to rush through a few of these, and I apologize, but I'd be glad to answer questions later. So this just shows a comparison of the MEA and the HCI results, and ultimately what it all boils down to is that overall, if you saw activity in the HCI assays, it was likely that the OP chemical would also be active in the MEA. Next slide, please. So now for our world and risk assessment, we work with external dose when we're doing our risk assessments, and we wanted to know, well, how does this in vitro data compare to our current endpoints that we use for OP human health risk assessment that are based on acetylcholinesterase? And to do that, we utilized in vitro to in vivo extrapolation to take that in vitro concentration and bring it out to an external dose, which we called an administered equivalent dose, or AED. And for this case study, we used high throughput toxicokinetics with a 3 compartment steady state model, which includes several assumptions that I can't get into right now, but the general approach has been published in many manuscripts by our colleagues at ORD and others. And we also had that approach evaluated by the SAP in 2014. Next slide, please. In some cases, we didn't have rat HTTK data, so we looked into whether we could utilize human data in the rat model instead. And this just shows an analysis of not just OP compounds, but all of the compounds where we were able to do this analysis to look at what would be the impact. And altogether, we went ahead and utilized the human data in the rat model in order to calculate these administered equivalent doses. And in those cases, we called it a humanized rat or HU rat model. Next slide, please. And so for the human comparison, since some of the assays were from human-derived cells, whereas others were rat-derived, we did different comparisons. If it was human derived cells, we used human-derived HTTK data with the human 3-compartment steady-state model. For rats, as I mentioned, if the data were available in the rat, we utilized that, but also we did this HU rat comparison as well to do the to see the impact of utilizing human data. And for 3 chemicals, we weren't able to do this. Next slide, please. And here are just a few examples. I can't show all of them since there was 27 of them, but ultimately, next slide, please. What we saw was that that some of the rat and HU rat AED values approached the in vivo rat BMD, BMDL that we saw in the in vivo studies. But altogether, they were either at or above values that we currently use for human health risk assessment. Next slide, please. I won't belabor this, but we also had PBK models available for some chemicals, and we saw similar results to the HTTK data or higher than what was predicted with HTTK. Next slide. Next slide, please. So as I mentioned, this is part of an international effort. So the ORD assays in conjunction with other assays that are being developed, particularly by EFSA-sponsored laboratories, can be utilized to cover all of the critical processes of concern for neurodevelopment. And if you just go one more slide to the FIFRA SAP, one more slide. As Anna mentioned at the beginning of this, we presented all of this to an SAP in September, asking about the ability of the in vitro assays to robustly evaluate the critical processes that they're looking at, and also how we conducted our AED to BMD value comparisons. And that should be available in about 90 days from the meeting. So we're looking at around December, and there is a link with that with much more detailed information about all of this, the assays themselves and our slide presentations that were provided. So I'm gonna go ahead and just skip 2 more slides. So you can skip over the conclusion. And I just want to mention that, go back one, for the how this fits into the NAM work plan is I think threefold, that we're trying to overcome the challenges and limitations in the in vivo DNT guideline study and utilize the data and information in a fit for purpose use. So the utilization of the data may change depending on the group of chemicals that you're looking at or the chemical. As I mentioned, looking at the reproducibility and the performance, and the case study, as well as our research products, establishes confidence and demonstrates application. And lastly, engaging and communicating with stakeholders like we have through the SAP, international collaboration, and forums like this. So my apologies for running over time. But I also
okay, so we'll take a couple of questions. So there's one, a couple of these will probably be better response on written anyway, because they're fairly technical. So Monique, did you learn anything? This is from Katie Walton. Did you learn anything about the mode of action of the OPs using the NAM batteries? Could you expand on this?
Currently, we still don't know what the mode of action is. And actually what we've seen is the chemical, there were very chemical specific reactions. You didn't see a trend necessarily for, the disruption of certain critical processes over others, that each chemical was very chemical, it was a very chemical dependent and results that we were seeing.
Okay, so we're already 11:45. I would ask you to respond to some of these in writing. There is one question from Francesca Pistolato about the times of exposure in the vitro assays. And it's highly likely that the group of the EPA staff that have access to these questions right now may not know the answer to that. So we may have to respond to this one in email form later on, but there are a couple more for you. But we're going to go ahead and thanks Monique for that. And everything that she's presented is also publicly accessible for those of you looking for more information. So we're going to pass it now to Andrew White, who is currently the science leader in computational toxicology at Unilever within the Safety and Environment Assurance Center, who's going to talk about integration of NAM data into work with cosmetics ingredients. So welcome, Andrew, and
Thanks, Anna. Yeah, just really to kind of reiterate what my fellow presenters have said, I'd like to thank the organisers for the opportunity to share some of the work we've been doing within a broad team over the last couple of years to kind of build our understanding internally on the use of NAMs in a kind of next generation risk assessment. Today, the talk really focuses on one out of several hypothetical case studies that we developed that were initiated, and in this case, this one was developed to integrate coumarin within a face cream. To start with, I want to kind of provide some background to set the scene and context of what we were trying to do, and then move into some more of the details of the study that was undertaken, and before hopefully kind of summarizing some of the learnings and outputs that have come through. So next slide, please. If we look at how this integrates really within the EPA non-animal approach, non-animal methodology work plan, I think really it focuses on the latter three areas, similar to some of the others have said. We set out to build internal confidence and understanding of the information, how we can integrate that, derive directly from non-animal methodologies through application and through a kind of realistic scenario relating to cosmetic use case. We not only utilized externally developed NAMs that were already available, but where we identified some critical information gaps, we then went ahead and extended that and developed some additional assays to integrate within that scenario, and we'll share some of that. And finally, as part of this workshop, but also it provided us with kind of case studies that we can then share those findings and the approaches that we've done, the thinking behind them, and engage with the community, our scientific peers, and also regulators about how we have gone about it and what we have found out. So to kind of gain feedback and further refinement for later on. So next slide, please. In our case, the stakeholders for this are primarily our consumers. And from them, there is an increasing demand that we for change in our approaches. They want products, yes, that remain safe and can be proven to shown to be safe in a transparent manner, but are not derived using animal testing. And increasingly, they want products which also meet sustainability requirements. So in order to kind of those three kind of key demands are very similar to what we see, or what I believe are similar to the goals that the EPA directive has been, and is also similar to the recently published EU chemical strategy. Both require change, both require innovation for safe and sustainable chemicals to be taken up and utilized, and both require us to adapt how we go about the approaches that we use for risk assessment in order to deliver that for our consumers. In the case of cosmetics, our overarching problem formulation can be described really by that simple, quite simple sentence: can we safely use X percentage of ingredient Y in product Z? It highlights, really from our perspective, the assessment is exposure-led, and has the advantage because it helps us narrow down the scope and enables us to progress, I think, quite rapidly in this space. We understand what the consumer use scenario is going to be, what product type it's going to be. We also understand how much of that ingredient is going to be used. So we have as I said, a narrower scope about what needs to be developed. If we go on to slide 3, please. And how we have gone about it is really shown, and the approach is shown at the bottom of this slide, in the bottom half. And it's an end-to-end, we presented it as an end-to-end approach, starting with exposure estimation and go through to the risk assessment conclusion. But actually it's derived from previous work that has been published over the years on frameworks that have been collated together. So we have used the tiering approach that has been produced in the Bregan et al. Paper, with some exposure-based weighting if it's feasible. We have taken into account, again, the tiering approach in the EPA framework diagram, but also incorporated the outline classification into non-specific or specific modes of action and incorporated that. And it follows the ICCR principles around next generation risk assessment, in that it's mainly human safety risk assessment focused. It's hypothesis driven, and overall it's a risk-based approach, as I previously mentioned, and it's designed to prevent harm rather than being predictive necessarily of the mechanism. While I have described it as an end-to-end process, it should be highlighted that in actual fact, it's much more of an iterative process, and it's flexible. As Clement said, we don't necessarily have defined assays. We should be bringing in assays based on what the information requirements are, and as such, what we are doing is, in an iterative process, taking the information from earlier findings and then applying them and saying where are our gaps going to be, and following up those with further studies later on. So if we go on to the next slide, please. So if we focus now on the kind of specific case study, the hypothetical example we have is we wanted to put 0.1% coumarin in a face cream. And to do this, we have made several restrictions on what data we can use, so that we ensure that we are not using any animal, any data that's been derived from animal methodology. So no in vivo data was defined up front. There was no history of safe use or any clinical data which would have been based on actually having prior animal data. So no use of animal data also from a read-across perspective, and any in silico alerts that were defined, which were based on animal data or from the structure of coumarin itself, were excluded, so that we were coming at this as if it was a novel chemical coming into it. And as I said, it's an exposure-led approach. Next slide, please. And just to kind of reiterate in a kind of schematic way, the approach that we are following, it's, as I said, risk-based, derived from a margin of safety that has been kind of previously highlighted by Rotroff et al. In 2010, where we are building out a case and understanding the consumer exposure and trying to get to a free plasma concentration, internal concentration, and then comparing that to points of departure derived from a range of hazard assays and biomarkers in order to see if there is the clear margin of safety that we have got free. So if we go on to slide 7. As I said up front, for us, we have a very clear understanding of what the consumer scenario is going to be, and we have access to publications, Hall et al., and databases around consumer-led habits and practices data. So from that scenario, we can define actually get a good reasonable idea of what the likely exposure is going to be. So we can define from a face cream, the amount of product used per day is going to be roughly about 1.54 grams at the 90th percentile. The frequency of use of that product is about 2 times a day, and so on, all the way through that. We have an idea of how much skin exposure there is and the exposure per duration of application, to get to a point where we can see what the kind of systemic level of exposure is going to be from an exposure. We then take that and have incorporated that into a PBPK model, in this case using GastroPlus. In the first instance, we have used ADMET Predictor to provide us with some in silico predictions of key chemical properties, and then taken those through and put those into the model and performed a sensitivity analysis to find which are the key parameters to follow up from an in vitro perspective. And in this case, it's we identified hepatic clearance, free unbound concentration, log P as clear parameters that needed to be followed up, and performed some in vitro assays to kind of refine the exposure based on those. If you go on to the next slide, this has then been shown in these 2 slides about the likely Cmax concentration that we obtain after running the PBPK model over a defined time period, in this case up to 10 days, to show that we get to a steady state level, both from an in silico and from where we had used in silico predictions for the internal clearance, or from where we got in vitro data that's come through. And then once we've got that steady state, we have used that as to try and derive a population model based on the variability, and you can see the spread of the distribution of the Cmax based on around that, and we use the 95th confidence interval in order to actually derive what that exposure is like. If we come back onto the next slide, please. Going into the hazard characterization aspects of the case study, where we could use existing data, we have pulled information together. So on the left-hand side, you'll see that we have performed some structural alerts and silico predictions based on the structure of the system using a series of different tools: ToxTree, MI Atlas, OECD Toolbox, also Derrick and Meteor to look at potential metabolite formation. And from here, we can see that we get some alerts for DNA binding, potential skin sensitization. The study I'm going to show you, or the information that follows, is really based on the systemic aspects of the case study. We have done some work on the skin sensitization, and I can put you in with that. Well, I won't explain today, but I can put you in touch with people who have more information on that. We had an MIE for genotoxicity, and we have followed that up, and potential for reactive metabolite formation to occur. From collating existing information, when we have looked, there were few active assays. We have used ToxCast data that was generated. We have looked into PubChem. We have looked at various different sources to try and identify any information that can be utilized to understand what assays were. And from here, we've got few that came back, but two of the key findings was from monoamine oxidases and carbonic anhydrases that actually had a concentration for point of departure between 3 and 40 micromolar, and that was taken forward into the margin of safety calculation. To return back to the MIE for genotoxicity, we have followed that up. We have utilized tox tracker assays to actually understand the likelihood of that, and that was done in both in the presence of S9 and minus S9. And as you can see, we have looked at both across DNA damage, p53 oxidative stress, and unfolded protein response. In each case, a positive result is shown in red and a negative in green, with some uncertainty where it's a potential is in amber. And for the positive +9, we clearly see that both assays for oxidative stress are positive, whereas without S9 we see no effects on DNA damage, and only one of the biomarkers for oxidative stress coming up. So in reality, from a coumarin is in the tox tracker assay is negative, but there is potential for some reactive coumarin metabolites that could induce DNA lesions, but probably secondary to an oxidative stress response is kind of the conclusion. If we move on to the next slide, please. We also performed, and one of the other areas that we looked at from an in silico alerts perspective was the MIE Atlas, which has been published by Allen et al. And Wendlake et al. Over the course of the last few years, which basically took a panel of 44 key targets, key assays that have been previously described by Bose et al. And are involved in drug attrition for those drugs where we can see it, and they cover off both GPCR panels, ion channels, enzyme panels, a whole range of different things. The predictions we got from the MI Atlas suggested there was no real alerts for any of those bindings, but following it up from in vitro assays using the Eurofins safety screen, again we saw a negative results for all these, with none of these assays hitting an induction level that is considered positive at 10 micromolar. Next slide, please. Beyond the safety targets, we then went and we've had a look at immunomodulatory effects caused by coumarin, and in this case we used the biomap diversity panel to investigate these possible effects. And again, we saw very limited effects when we looked at dose response profile. In the graph you can see here, we can see the 8 different panels of primary human cells that have been grouped together. And what we were looking for is data that went above and outside of the background noise associated with the assay, which is in the grey. And the most sensitive assay we got through was for these 3C endothelial panels, and it was an anti-proliferative effect. And 18.5 micromolar was the low L that was obtained from this. But again, above what we expect to see from a consumer exposure scenario. So data suggests, again, it has no immunomodulatory effects at the relevant concentrations, and it's not an anti-inflammatory compound in this case. If we move on to the next slide. Where we wanted to go beyond just looking at defined modes of action, we want to cover off the ability for where compounds are showing non-specific biological activity. In order to do that, we have developed with collaborators kind of cellular stress response assays that cover off 10 key cell stress pathways based around the Simmons et al. Paper for 8 pathways, and then adding in some mitochondrial toxicity and some cell health biomarkers. And we have various biomarkers within each of those 10 pathways that were followed through. Each case, we have done this at various, formed a dose response curve and done these at 3 different time points, primarily in HepG2 cells to start with, but then extending out to NHEC cells. The approach we have followed for doing this has been published in the Hatherell et al. Paper, and there is more information there on the assays and what their findings. Also, we have utilized a Bayesian a statistical approach to define dose fitting for this and a point of departure. This approach uses provides a confidence score on dose response, and we then use a point of departure, which is 2 standard deviations above background for each of these assays. The data you can see here is shown for HepG2 at the top, and then for some of the NHEC cells below, and relates to at any particular time point any of the assays that had a confidence dose score of greater than 0.5, suggesting that there was a dose response, and where the point of departure was for each of these. And you can see in most cases these were quite high, up at the 700 micromolar sort of range, in the hundreds of micromolar range. But then for the mitochondrial toxicity, some of them came down at lower concentrations. And here you can see the confidence dependency score, concentration dependency score that was for each of these, which gives you a kind of an idea of the robustness of that concentration response. So these were quite high concentrations for where we were getting positive hits for the coumarin. And if you look at the next slide, please. Just to kind of put that in perspective, when we look at the HepG2 responses and then compare it against another panel of compounds, you can see that we see there is not very much activity compared to other compounds where there is clearly much more bioactivity from a drug perspective or something like that. So we're not seeing much bioactivity going on in these. Next slide, please. Just to kind of and finally, just to kind of ensure that we are trying to get as much of the biological coverage as we possibly could, we followed this through with some high throughput transcriptomics, and in this case we utilized the TempoSeq approach that has been previously described. We have used the human gene panel version 1, which was the 21,000 full panel in this case. And we did this on 3 different cell lines just to kind of give us a broader coverage. So we'd used MCF7 cell lines, HepG2 cell lines, and HepRGs in the first instance. And that gives us a bit of indication of metabolism that's in there. And you can see there are differences that we obtained within this. In essence, pretty much in terms of the number of pathways, number of pathways that were hit as active. We have utilized, in terms of the analysis approaches, we have used the BMD Express 2 software that has been developed and come out from Scott Arbach and group. And then use the in order to actually understand how to apply that from a pathway test, we utilized the paper that had been published by Farnahan et al. Earlier, where they had compared 11 or 12 different methods for aggregating transcriptomics pathway and compared that back to some apical endpoint data as well, to try and understand which ones were closest matching. We have taken 5 of those approaches through, and the data you're seeing on the right hand side is the point of departure derived from each of those different approaches, and the ones in red are ones we have taken through into the analysis. So from the BMD with the lowest reactome pathway with the lowest BMD, these are the ones which we've taken through from HepG2 and HepRG2. The MCF7s in this case, we saw very little robust signal that came through, and could not derive a pathway to point of departure from that. and the data was not very robust, and we, in this case, we put that to the side. If you go on to the sorry, can you go back one, please? Just to finalize, we have also started, just to extend this, utilizing another approach, building on the Bayesian work that had been done for the cell assays. We have transferred that same technique and extended it into the transcriptomic space to make it more bespoke, and that has provided us with a couple of I think potential advantages where we can extend this going forward. It provides us with a kind of distribution around where that point of departure is likely to be. It focuses on the question of at what dose, what is the highest dose where we don't see, what is the lowest dose we don't see any changes or perturbations that meet the confidence determined score of greater than 0.5. And the assumptions that are built into it are clearer, so that we're not having to make a series of assumptions in how we define thresholds, as we're doing in the benchmark dose express software. And that approach has now been accepted for publication, has been put out in ComTalks recently. Next slide, please.
Andrew, there's 2 more minutes, please.
Yeah. So just before completing all of those and sharing margin of safety, one of the key aspects where we did have some uncertainty and where we did want to go back and refine the process rather than just relying on the alerts that we've got was around the metabolism. And in this case, we've gone back through and looked in terms of the metabolic pathways and what the likely metabolic components are that we can actually pick up. So we've gone down and done some metabolic identification. And in this case, most of what we were seeing was derived from one metabolic cytochrome, and the primary pathway that we were seeing until we get to very highly saturated levels was coumarin was detoxified to hydroxycoumarin and then on to secondary metabolites after that, so not the epoxide formation which would drive the reactive alerts. And this is further supported by data where we were using 3D HEPA-RGs to try and induce and increase the amount of metabolism in some of the assays, and we've done this on the HTTR high throughput screening, high throughput transcriptomics assays, and again the points of departure we were getting were in the same range as what we were getting within the other assays as well, suggesting that the higher metabolism was not driving any more lower points of departure for what we're seeing. Next slide, please. And this just summarizes all of that information into one overarching graph where we are seeing what we're showing really is that the Cmax values where we are looking at internal exposure were lower than all the points of departure, and the margin of safety in each and every case was greater than 100 from where we were looking at the points of departure for the bioassays that we performed. Coumarin in this case was not genotoxic. It did not bind to any of the targets and does not show any of the immunomodulatory effects within those exposure scenarios or relevant exposures. And the weight of evidence would suggest that under these circumstances, the outline is that it would be safe for consumer use. If you can go on the next slide, please. So just in summarizing, the focus of this was about integration. It was about building lots of different lines of evidence, weight of evidence to show how the tools can be made to make a safety decision, pull it all together rather than relying on one or two assays. A key part of that is that it requires diverse expertise. We pulled together a great team to actually do this. The exposure-led approach is really around that protection goal, risk-based protection goal. We're not really out there to define what the mechanism was, but to define, are we protective of human health and what the margin of safety is that's required to do that. its strength is coming really from a combination of targeted and unbiased tools, kind of broad biological coverage, rather than any one of them. We have to do further in that. You know, this is not the end goal. This was a first scenario case study. We have followed up where we think there is uncertainty, and you can continue to refine that. We think there is further evaluation needed. We are doing additional case studies. This was the first one, both internally, but also in collaboration. We have an ongoing collaboration with the EPA where we can hope to broaden these and do a lot more of the chemicals, and also within EU TOTS risk, another a group where we're involved. We are building out additional case studies so that we can build the weight of evidence and the understanding of how to apply these in a broader context. We realize there is still gaps in this, and there is additional research ongoing to kind of build out and progress on those gaps. So next slide, please. And I'd just like to say, as I said, this is a broad group of people with various expertise, and there's been a lot of work gone into this. And I just like to pull out Maria Baltazar, Alison Middleton, and Joe Reynolds as kind of some of the key people who have been building out. Alistair's been building out the cell stress panel. Maria was the team leader who ran the case study, and Joe has been building out some of the statistical approaches that we have been applying and developing. Another one is around on the exposure aspects. Tom Moxon has published that as another paper. I'd like to thank you for your time.
Thank you, Andrew. I appreciate the presentation. That was really interesting overview and showing sort of the framework that you used and how you adapted that. There are a few questions in the Q&A list for you, but we're actually at time. So I was trying to find one that would be a quick answer, but they all seem very somewhat overarching. There's one, if just sort of a quick response, if you can, on given differences in the POD from the different cell lines that you showed, could you tell us your criteria for selecting the cell lines you used?
Yeah, I mean, there was some pragmatism in those cell lines. Some of those cell lines were used because it will benchmark against other work that's ongoing. Some of those cell lines were used because they matched other assays that we were doing at the same time. And we extended it out because we wanted some cell lines where there was some indication of metabolism. And we realized that they're not 100% on the metabolism, but it gives at least an indication of what's potentially to go on.
Okay, great, thank you. And like I said, there's a few other questions in the Q&A, and there's something odd going on with the Q&A where they're not all being time stamped. So there's some below what you're looking at. If you want to dig through, they're assigned to you. So hopefully it'll be clear. Otherwise, we'll follow up with questions later. But thank you very much. I appreciate it. Now I have the opportunity here to present the last and final case study, number 3, on incorporating the threshold of toxicological concern into regulatory decisions under the amended Toxic Substances Control Act. Dr. Todd Stedderford will be presenting this case study for us today. He's currently a senior science advisor for the Office of Pollution Prevention and Toxics, or OPPT, at the US EPA. Todd?
Okay, thanks. Next slide, please. Okay, so today I'm going to cover the Toxic Substances Control Act, or TSCA, and specifically about regulation of new chemical substances under Section 5 and existing chemical substances under Section 6. Also touch upon Section 4H of TSCA that deals with the reduction of testing on vertebrates, and thereafter I'll talk about the threshold of toxicological concern, or TTC, some of the background on that, some examples of where that's currently used by regulatory agencies, and also some potential areas where that could be used under TSCA. Next slide, please. So this slide, it provides really just an overarching view of new chemicals and existing chemicals and how they're regulated. But just as some background, TSCA was passed in 1976. It's the US's primary chemicals management law. And as a lot of people are aware now, on June 22nd, 2016, the Frank R. Luttenberg Chemical Safety for the 21st Century Act was signed into law, which amended TSCA. And TSCA gives EPA the authority to regulate chemical substances, which means any organic or inorganic substance, but not mixtures or pesticides or tobacco products or things like food additives, drugs, cosmetics, or medical devices. So, one moment, let me shut my phone off. Sorry. So new chemical substances, those are compounds that are not included on the list of chemical substances compiled and published under Section 8B of TSCA, and that's also known as the TSCA inventory. So if a chemical is listed on the TSCA inventory, it's considered an existing chemical substance. And the TSCA inventory, it's updated about every 6 months, and it contains more than 86,000 chemicals, of which about a little over 41,000 are active, meaning that they're being manufactured, introduced into the stream of commerce. So on new chemicals, TSCA sets forth 5 possible EPA determinations that can be made. You can see that on the left side. So the first is the chemical presents an unreasonable risk of injury to health or the environment. The second being the available information is insufficient to allow the agency to make a reasoned evaluation of the health and environmental effects associated with the substance. The third is in the absence of sufficient information, the chemical may present an unreasonable risk of injury to health or the environment. The fourth is the chemical is or will be produced in substantial quantities and either enters or may enter the environment in substantial quantities where there is or may be significant or substantial exposure to the chemical. And the fifth is the chemical is not likely to present an unreasonable risk of injury to health or the environment. Now on the right side, the existing chemical space, as I said, those are the chemicals that are listed on the TSCA inventory, so under Section 8B. EPA is required to prioritize existing chemical substances as either low priority substances or as high priority substances, and to perform risk evaluations on those substances designated as high priority substances. So EPA defined what a high priority substance is as a chemical substance that EPA determines without consideration of costs or other non-risk factors that may present an unreasonable risk of injury to health or the environment because of a potential hazard and a potential route of exposure under the conditions of use, including an unreasonable risk of potentially exposed or susceptible subpopulations identified as relevant to EPA. Next slide, please. So review of new and existing chemical substances at EPA are risk-based determinations and evaluations. They must be based on the best available science, weight of the scientific evidence, and without consideration of costs or other non-risk factors. So EPA assesses a wide variety of potential human health and environmental effects associated with the known, intended, and reasonably foreseen conditions of use for both new and existing chemical substances. Next slide, please. So the types of data utilized for new chemical substances, new and existing chemical substances. We use, submitters are required to provide all data in their possession or control related to health and environmental effects for new chemical substances. And we may require the development of new information relating to the chemical substance. And one thing about TSCA is that there is no upfront testing requirements under TSCA. So if somebody wants to manufacture a new chemical substance, they don't have to go out and perform a bunch of testing. They can basically submit the required information under the statute with chemical ID, manufacturing process, and stuff like that, uses, but there's no upfront toxicological testing. For existing chemical substances, EPA must first take into account reasonably available existing information, and EPA may then require the development of new information relating to the chemical substance. Next slide, please. So the TSCA chemical hazard approach, the data preference for TSCA risk assessments, it includes chemical specific data followed by analogs and then modeled data. For analog data, EPA considers the physical chemical properties, the presence, position of reactive functional groups, potential metabolites, degradates. EPA also uses the TSCA New Chemicals Program chemical categories document and structural alerts to identify hazards. But the question is, what if there's insufficient information for making a risk determination? Next slide, please. So the amendments to TSCA in 2016, they included statutory mandate for reducing tests on vertebrates. So prior to requesting testing on vertebrates, EPA must consider reasonably available information and encourage and facilitate scientifically valid test methods and strategies that reduce or replace the use of vertebrate animals while providing information of equivalent or better scientific quality and relevance that will support regulatory decisions. So next slide, please. And that brings us to the threshold of toxicological concern. And one thing I forgot to mention, I have footnotes listed on the slides. If you go to the notes pane, once the presentations are made available, I provided full citation information and URLs to the underlying information if you'd like to read up further. So the TTC approach, or threshold of toxicological concern, is possibly an approach that could help fulfill Section 4 requirements. And we'll get into some of the more specific aspects where this could be actually employed. So, but other agencies have looked at this. So you have the European Food Safety Authority and the World Health Organization. They identified the TTC approach as a pragmatic, scientifically valid methodology to assess the safety of substances of unknown toxicity found in food. But what is the TTC approach? So it's basically a level of exposure that is considered to be of no appreciable risk to human health, despite the absence of chemical-specific toxicity data. And I included a link there. There's a really nicely done presentation on this topic that's freely available if you'd like to read up more on that. Next slide, please. So TTC approach, it utilizes the classification scheme originally proposed by Kramer et al. In 1978, which consisted of a decision tree of 33 questions that lead to a final classification of low, moderate, or serious toxicity, so class 1, class 2, or class 3. And in 1996, Monroe et al., they evaluated a database of 613 chemicals. They classified the chemicals according to the Kramer classes. And if you look on the lower left side of the slide, they calculated the cumulative distributions of the no observed effect levels for each. And from this, they derived human exposure thresholds by multiplying the 5th percentile NOEL by 60 kilograms, which is the assumed adult body weight, and then dividing by a safety factor of 100. And that's where they came up with the human exposure threshold. So for Kramer class 1, as an example, they came up with 1.8 milligrams per day. Next slide, please. And subsequent work by Croze et al. In 2004 led to the development of a proposed decision tree that's shown on the right that incorporated different TTCs to different structural characteristics. And it's hard to read that on there, but you can see some of the questions. Does it raise concern for potential genotoxicity? Does it exceed the intake of 1.5 micrograms per day? Is it an organophosphate, and so on? Next slide, please. So the TTC approach, it's a tool that may be used for justifying waiving the generation of animal data and is regarded as an extension of read across and chemical category approaches, which we currently use under the TSCA program quite extensively. It's been used by regulatory agencies. Two examples listed there for the U.S. Food and Drug Administration and the European Food Safety Authority when considering very low-level lifetime oral exposures. Next slide, please. So just some background on FDA's approach. In 1995, FDA implemented a threshold of regulation value of 1.5 micrograms per person per day for food contact materials. And the TOR applies to substances that migrate or are expected to migrate into food and are exempted from regulation as food additives at levels below the TOR. And then there's a series of requirements that it's not shown to be carcinogenic, no reason to believe it is carcinogenic. The substance doesn't contain carcinogenic impurities, and if it does, it must have a TD50 value less than 6.25 milligrams per kilogram body weight per day. The TD50 is the dose that causes cancer, 50% of the test animals when corrected for tumors found in control animals, and so on. Next slide, please. So collectively, the research by Monroe, Crows, EFSA, and others led to the current TTC values used by the European Food Safety Authority. So they have cutoff values for potential DNA reactive mutagens, carcinogens, organophosphates, carbamates, and then the different Kramer class 3, class 2, class 1. And just to note that the overall workflow and data sets for the Kramer classes are not the same as the data sets used for substances with genotoxicity alerts, nor for the organophosphates or carbamates. Next slide, please. So there are some exceptions here. So EFSA identified the following substances as not suitable for the TTC approach. So those covered by regulations that require the submission of toxicity data, those with data that allow for a risk assessment. As I mentioned under TSCA, a lot of times we will use analogs. So, we do have that option. Those which are not represented in the data set or outside the domain of applicability, so inorganic substances is an example, and those with special properties like high potency carcinogens, steroids, and substances with the potential for bioaccumulation. Next slide, please. So the TTC approach, it's well established for oral exposures. However, TTCs have been proposed for inhalation exposures, for route to route extrapolations, other endpoints, including like skin sensitization and for ecotoxicity. So the question is, has the TTC approach been considered for regulatory decisions on industrial chemicals? And the answer to that's yes. There's several examples of where regulatory agencies have used the TTC approach for industrial chemicals. So Health Canada, EPA, and EPA. Next slide, please. So Health Canada in 2016, they identified 89 substances from their list of priority substances which were unlikely to pose a risk to human health using a TTC-based approach and current exposure levels. ECA, in some of their guidance under REACH, discussed different TTC approaches, their limitations, the criteria for use, and their potential use under REACH. So REACH is the law in Europe that's comparable to TSCA in the United States. And ECA provided a generic TTC scheme that illustrates the way that TTC may be used and could precede any substance-specific testing. Next slide, please. So I took this figure, directly from the ECA guidance, and it's, as it shows, a TTC approach, it would precede any chemical-specific testing. There's one tier shown, but one could apply additional tiers as illustrated in the decision tree that I showed for Crows et al. 2004. And basically, again, this is looking at, are your exposure estimates lower than the TTC value for the chemical substance? Next slide, please. So EPA has also considered this in 2018 when they issued their draft strategic plan. The TTC approach was identified as a possible evaluation for its utility, with priority setting, with expanding the existing new chemical categories, identification of new categories for the new chemical categories document. And in EPA's response to comments on that draft, they stated that they believe that exploration, and implementation of the TTC approach, at least for some structural classes, is an important possible avenue for some TSCA decisions. and EPA is considering this topic as part of collaborative efforts. Well, the collaboration was initiated in 2020 by external stakeholders with EPA and other groups with the intent of exploring potential TTC applications under TSCA. Next slide, please. So now I just want to cover some of the aspects for new and existing chemicals where this could actually fit in. And I think it's important to note, like some of the areas under TSCA where we already have a built-in TTC-like approach. So EPA implemented this as an exemption for some new chemical substances, and it's regardless of the production volume. So in 1995, EPA added a new exemption category for new chemical substances with low environmental releases and low human exposures. It's known as the low release and exposure exemption, or low REX. And the exemption was developed based on EPA's experience conducting risk assessments on over 25,000 new chemical substances up to that point. Next slide, please. So the low REX exemption, it provides several generic release and exposure eligibility criteria. So for surface water, direct and indirect releases are expected to be prevented with a limit up to 1 part per billion. For ambient air, the maximum annual average concentration from incineration is 1 microgram per meter cubed. For releases to land or groundwater, no releases are expected for landfill or other land disposal methods unless the submitter shows that the substance will not migrate to groundwater. And there's some testing recommendations in the Federal Register notice if you'd like to read up more on that, that I cited on the previous slide. Fugitive air, unrelated to incineration, EPA did not provide a generic eligibility criteria for this, but it noted that emissions under 23 kilograms per site per year were seldom found to present an unreasonable risk. And for exposures, there's no dermal or inhalation exposures permitted to workers, and that does take into account the engineering controls, work practices, and personal protective equipment. And for consumers and the general population, no exposures are permitted other than the eligibility criteria stated above under the releases. And upon approval of a low REX exemption, the uses and exposure release controls are all binding, and as are the listed manufacturing sites. And these can only be changed if specific conditions are met. Next slide, please. So EPA excluded new chemical substances with possible consumer and general population exposures from the low REX exemption. And the basis for that was that EPA concluded that they could not develop meaningful exposure criteria that would consistently screen out those substances which may present unreasonable risks through the wide range of possible exposures. So if a submitter's planning to submit a new chemical substance and there are intended consumer uses, those could be paints, detergents, dyes, and just lots of other examples. The submitter would have to consider submitting the chemical substance as a pre-manufacturing notice or under another PMN exemption. Another one that's available is a low volume exemption, or LVE, which I'll talk about on the next slide, please. So also in 1995, EPA implemented an exemption for the new chemical substances manufactured at 10,000 kilograms or less per year. And LVE substances, including reasonably anticipated metabolites, environmental transformation products, byproducts, impurities, they will not be approved if they raise serious acute or chronic health effects or significant environmental effects. And the proposed controls for exposure release are binding on the manufacturers, as are the uses and manufacturing sites, again, unless specific conditions are met. And LVEs are only approved if EPA affirmatively finds that the manufacturer, processing, distribution of commerce, use, and disposal will not present an unreasonable risk to human health or the environment. Next slide, please. So for new chemical substance notification, so that would be like a standard pre-manufacturing notice or a significant new use notice. EPA and submitters, they generally identify chemical analogs for informing the risk assessments, and they can also do that for the like the low volume exemptions too. But when no suitable analogs are available, the TTC, it may be used by submitters to identify, for example, opportunities for exposure release reduction through engineering controls, workplace practices, and or personal protective equipment prior to submitting a PMN. However, prior to utilizing that approach, we would encourage submitters to consult with EPA using the standard mechanism for that. It's a pre-submission meeting, which can be requested through the URL shown on the screen. Next slide, please. So opportunities to incorporate the TTC approach for existing chemical substances under TSCA. So touched on this previously under Section 6 of TSCA and the implementing regulations for prioritization and risk evaluation. The EPA is required to screen existing chemical substances against a bunch of different criteria. Just read a couple. So hazard and exposure potential, persistence and bioaccumulation, and so on. And based on the results of the screening, EPA proposes to designate a substance as either a high priority substance or a low priority substance. And EPA considers additional information collected from the proposal, and then they publish their final designation for each substance. And again, if something's designated as a high priority substance, EPA has to perform a risk evaluation. Next slide, please.
Todd, 2 more minutes, please. Thank you.
Okay, I'm almost done, thanks. Okay, so opportunities. So Dr. Patlowitz and Dr. Wambai, they published an approach that it used chemical-specific high-throughput exposure estimates and the TTC approach to prioritize thousands of substances. And the results indicated that this combination of the HTE-TTC approach could be adopted as a pragmatic first step in risk-based prioritization of thousands of chemicals. And if you recall, as I mentioned, the TSCA inventory of existing chemicals, it's got over 80,000 chemicals, and about 41,000 of those are active. So you really need these pragmatic approaches for identifying the ones that really should be evaluated. Next slide, please. And conclusions, TTC approach, it represents a possible tool for screening some structural classes of new and existing chemical substances to make preliminary decisions. Examples would be evaluating exposure release controls for new chemical substances and identifying the need for possible refinements in the absence of chemical specific or analog data for risk-based prioritization of existing chemical substances, and as a non-animal approach for making initial decisions regarding the need for further data gathering. Several points warrant further evaluation. Oral TTC values are well established. However, TSCA risk assessments may include evaluations via other routes, so dermal and inhalation, other endpoints. So rather than systemic, you could have portal of entry effects. You can have less than lifetime assessments, depending on the conditions of use, ecological assessments, and so on. So next slide, please. And progress continues to be made in this area. So Nelms and Patlowitz just published a proposed inhalation TTC approach that's in press. Eco TTCs with subcategorization into MOA-like categories is underway with the HESI project as an example. There's been refinements to the organophosphate and carbamate alerts. Nelms et al. 2019 delve into that. There's been work on internal TTCs, and there's also online resources like Citovation's TTC Data Mart, which includes TTC values that were established for something like 45,000 chemicals. Next slide, please. I'd just like to acknowledge the contribution from my colleagues, Kyle Henry, Gino Scarano, Iris Camacho, Grace Patlowitz, and also a special thanks to Rick Becker for providing feedback from ACC on this. And that's it. Thank you.
Thank you very much, Todd. I appreciate it. Thanks for that overview and the overview of TSCA, but also the TTC efforts and integrating that into TSCA. We have a few questions here. Some might, I'll go through them in the time that we have. We have a few minutes here. The first is, how do you define a scientifically validated test method? Is TTC used in TSCA high priority substance regulation? So,
well, we haven't used it yet. It's I was just trying to illustrate some ideas where it could be used. I think that this is something that we're going to delve into through that collaboration to see, you know, is it really a fit for purpose NAM that we could potentially use under new chemicals or existing chemicals.
Okay, and then the first part, I blurred them together a little bit too much, but how do you define a scientifically validated test method? And I don't know if there's an official EPA definition that you're aware of, or...
I was just thinking, is there really one? I'm not sure.
We can follow up on that part if there's one related to TSCA that you're
I will
using.
say though that I've mentioned this in other meetings with ICVAM, where a method doesn't have to be validated for somebody to propose actually utilizing it for like on the new chemical space. If they want to utilize something, we will look at the data, but before embarking on something that isn't, you know, like an OECD test guideline or something like that, it is best to have that pre-notice consultation meeting to discuss, you know, what's being planned and also how we could actually utilize that data in a quantitative risk assessment.
Okay, thank you. And I'll go, this next question is from Jennifer McPartland, which sort of ties into part of the answer you had before, is where can we get more information about the 2020 collaboration focused on TTC under TSCA?
Well, I think that's all the information that's available now is what I had stated. It's really going to be a collaboration with, it's going to be external partners. It's going to also rely on our experts in the Office of Research and Development and you know, the core TSCA experts that we have here in OPDT. So, but I don't think this has been, I don't think there's been much much in the way of you know, really fleshing out the details for that thus far. It's just, you know, it was just the initial introduction and, hey, you know, let's have this collaboration, let's have this discussion and see what we can come up with.
So the follow-up from Jennifer, so it hasn't started yet. You're just in the initial stages, it sounds like.
Yeah, it's really the initial stages. We have a potential, you know, draft list of topics that we want to cover, but we're not going to have a kickoff meeting, I believe, until sometime next month. So this is definitely in the early stages at this point.
Okay, thank you very much. And there are a few other questions for you in the Q&A box. If you can address those here now, we can post them live, or we'll follow up through the email, as we said before. So thank you very much for your presentation, and thank you to all the speakers today. I know I'll speak on behalf of Anna as well, but thank you for your presentations, and thank you to the audience for your engagement and the questions we've received so far. So I have the pleasure of introducing for our closing remarks and to close out the workshop today, and for the full workshop, Mr. David Dunlap, who is currently the Deputy Assistant Administrator for Science Policy in the Office of Research and Development here at the US EPA. He has over 30 years of environmental health and safety experience. And since joining the agency, much like Alex, Jennifer, and the administrator, he's been very engaged in many of ORD's and the agency's science initiatives, particularly looking at incorporation of NAMs and reducing animal testing by the agency. So David, I'll hand it off to you to bring us to a close. Thank you very much.
All right, off mute. Thank you, Maureen. I'm going to have to modify that bio. I like to feel like I have a lot of experience when you say 30 years of experience, but I don't like everybody know how old I am. But thanks anyway. Thanks. That's my fault. But good evening, good afternoon, good morning. Regardless of your time zone, thank you everyone for participating in this second annual State of Science Conference on using new approach methods for chemical safety testing. It's been said a couple times, but I want to thank my colleagues in OCSPP and ORD for coordinating this workshop, our speakers, our presenters, the moderators, and all of those who've asked questions and otherwise provided discussion items. and as well our ICF contractors for all their support. You know, frankly, if it wasn't for them guiding us through here and making sure that I pressed the right button and was off mute, you know, most of us would not be able to be capable of maybe, you know, moving through the Zoom conference as flawlessly as we do. So we are aware of all the thousands of chemicals in commerce today, and how more information on these chemicals would help us better assess those potential risks. That is why the research and development of the use of NAMs is so critical. When we identify critical data gaps regarding chemical toxicity exposure, NAMs will help us rapidly develop that important information that's key to protecting human health and the environment, all while using fewer resources and decreasing our reliance on animal testing. When properly refined, NAMs will fundamentally change the way EPA evaluates the toxicity of chemicals and will ultimately improve our ability to respond not just urgently, but with even greater certainty to the public's questions about the safety of a chemical. This conference, the EPA NAM work plan, and the deliverables within the plan are critical steps towards achieving those goals. Earlier today, we heard about the current state of NAMs research and the application of NAMs in decision-making. From advancing the state of the science to increasing our confidence in the application of the science, it is clear we have made significant progress, but it's also clear we have more work to do. I'm quite inspired by the conversations and presentations I've heard over the past 2 days. And I am confident that we will meet the administrator's goal of reducing animal testing while improving the quality of the information and the confidence of the decisions that use this information. As we think about the work ahead and the challenges to overcome, let's not forget about the great work we heard about during this conference. For over 2 decades, EPA's Office of Research and Development has been conducting innovative NAMs research. We made significant strides. As you heard this morning from Chad, ORD is developing methods for incorporating metabolic competency into high throughput screening assays, which many have cited as a weakness of NAMs. You then heard about a few case studies which are using NAMs to evaluate chemicals. Monique discussed how ORD has been working closely with OCSPP to develop and use NAMs to evaluate chemicals for potential developmental neurotoxicity. In addition, we heard from Andy with Unilever, describe a case study done in collaboration with EPA researchers to evaluate how to use NAMs for assessing the risks of chemicals found in cosmetic products. Finally, we just heard from Todd about how OCSPP is using NAMs to incorporate thresholds of toxicological concerns, TCCs, into risk assessments. In looking to the future, I'm pleased to say that EPA continues to make great strides in our new approach methods work, and I'm looking forward to the results of those research efforts and sharing them with all of you. The NAM work plan is a living document that the agency plans to update as science advances and application of NAMs evolves. NAM's research and development will rely extensively on collaborations across governments, such research organizations and industry. So we all encourage you to submit your feedback on the work plan and tell us how you can help EPA achieve these goals. I also encourage your feedback on the conference format and content. This is only our second NAMS conference. How can we make it more informative? Should we reorganize in some fashion? Should we have breakouts for specific areas of science? Of course, we are virtual this year and we all hope to be in person next year, but just provide your feedback, keep that all in mind as you do that. I think that will be very important. We look forward to your input on the science and the work plan, but I think communicating our activities, our progress is just as important, especially in these days and times. So all input will be welcome, and you can do that by emailing us at nam@epa.gov. I'm sure that's printed a number of places. You've heard that numerous times today. We plan to hold a State of the Science conference annually. The next one will be held right around this time next year. We will be 1 year close to our milestone of reducing the use of animals by 30% by 2025. And so I anticipate having plenty of interesting science and good news to report then. In the meantime, please check the EPA NAM website regularly for updates, and that's epa.gov/NAM. I look forward to continuing to work on all of these issues with each of you, and please take care, and hopefully next year we can meet in person. Thank you.